So you would have to measure there. You have to do something like that, like 100 meters out that way, and then.
That would be perfect.
So
when I push the button to start the transit, that's when it actually marks the time. So I have that minute to put it in.
Every time we go to time signature.
All right, we're back up.
Okay, we're back up. We're getting ready to do a transect. A transect is we follow a direction at a specific speed. Here is 0.1 meters per second and 1 meter from the bottom. And we have the laser beams, so that way we will move along this line and we'll be able to count all the organisms. And here we not only know what is there, but how many of each one. Ahora nos estamos preparando para hacer un transecto. Y un transecto es una ruta que seguimos, una ruta fija a una velocidad fija, que en este caso es 0.1 metros por segundo, y a una distancia fija del fondo. En este caso es 1 metro. Y tenemos los apuntadores láser que marcan 10 centímetros entre uno y otro. Y eso nos da una medida y entonces podemos cuantificar qué es lo que está ahí. Entonces no solo sabemos las especies que están, pero también cuántos hay de cada una de las especies. Y eso nos permite comparar diferentes regiones.
So am I going to have the ship? They're going to follow us. Want to take that? Or they're going to go straight through?
we follow us at that point. If they stay on the line, then we're going to be in the line, so we'll see how it is.
Just check that as we go.
So go.
Just tell me what you need, Mike.
So I don't know how to do like lines though between those markers.
And we do go, yes, 0.1 meters per second.
Or at least if you draw the first line to the first marker, so you kind of follow with this to measure. Just measure from where we are to the first marker. And I'll just keep that line. Yeah. Look at there.
Happy with that?
Yeah.
Where do we see the altitude?
And the altitude is
From
just here.
from the bottom.
Yeah, so we're up like 10 meters just now.
Okay. So we're gonna go
But
down.
we're not going to see the meter because obviously it's going down where our altimeter is. It's measured in maybe 3 meters, but we could be camera-wise pretty close.
Okay. When you have it in position, let us know and we'll start. Yeah, we're getting in position to start our transect.
Happy with that sort of picture?
Yeah, looks good. Looks good. Okay,
Good. I'm happy to start.
okay. Start transect.
Right now?
Yep.
Okay.
And let's go. 100 meters.
The lower line, speed of ground 0.1.
Here we're moving at 0.1 meters per second
Heading to 9.
and at a meter above the bottom. And as I mentioned with the laser beam, so we will be able to quantify what is there.
0.1 what?
we're looking at different species of octocorals, all the way from the long fingers, like we're seeing here, to very large sea fans, to little ones, the green on the bottom. So you can, just from here, you can see how diverse this is. And also we're seeing small sponges, different species of sponges, some tube worms. And ya estamos haciendo el transecto y aquí vamos viendo diferentes especies de octocorales, algunos muy pequeños como unos verdecitos que vimos, otros que son como látigos largos y también vemos unos abanicos, algunos de esos calculamos que pueden medir casi 1 metro y una diversidad de esponjas que podemos ver en estas imágenes, desde unas que parecen pétalos hasta otros que parecen copas. Y también vemos organismos como ese uno transparente que está ahí, eso es un tunicado, que está relacionado a nosotros. Ahí vemos una esponja vieja, probablemente muerta ya. What we've seen here is many sponges, and that one that looks brown is maybe dead. We're not very sure. There's many surprises down at this depth. There's looks like cup coral, but maybe not. Maybe it's anemone. That feathery fin on the bottom part, that's a crinoid. That animal that look transparent, that's a tunicate. It's a group of animals that are relatively close related to us as human beings.
Bridge oil, we set a new waypoint, and it's zigzag on this face. New waypoint, zigzag. That's
Behind me is 2 other persons, Janet and Rob, and they help me mark everything that we're doing, so we know exactly where we started the transect, where we finish, and they keep notes of the different organisms that we're looking at. And because when we're doing a transect, we cannot stop, then we mark the site and come back to do collections if that's of interest. Aquí conmigo están Janet y Rob, y lo que hacemos es que ellos van apuntando, si vemos algo muy interesante, queremos volver a verlo, lo marcan, porque cuando hacemos el transecto vamos sin detenernos en la misma dirección siempre para que se siga una línea relativamente recta. Y aquí vemos más octocorales, estrellas frágiles, pepinos de mar, eso rosado, anaranjado, es otra especie de octocoral. En la parte de arriba vemos una esponja. Right here we're seeing a large, very large sponge with little stars and many different species of octocorals. And if you notice, there's animals living on top of the octocorals. So we call this engineering species, or like the sponges, that they create a structure where other organisms live, like the building where other organisms can live. Here is a different type of sponge, that globular sponge. Yeah, the sponge, mark that, please.
This is another type of coral.
Aquí seguimos viendo diferentes grupos de octocorales, diferentes formas de esponjas. Acabamos de ver una esponja globular. Estas otras son más como pétalos de flores. Otros son como costras sobre las rocas. Que estamos viendo en la parte superior una de las esponjas amarillas que descubrimos ayer. Un color muy brillante. Y les recuerdo, aquí estamos a 3.000 metros de profundidad. Eso significa que no hay nada de luz. La luz penetra como unos 200, poquitos metros. Y además la temperatura allá afuera es como 1.7 grados centígrados. Entonces es frío y oscuro. Pero con las luces artificiales del vehículo podemos ver toda esta maravilla. So I remind you that what we're looking at is 3,000 meters below the surface of the ocean. So it's pitch black. The light penetrates only a couple hundred meters. And what happened? Okay, lost the signal for a second there. So as I was saying, this is pitch black. Light only penetrates a couple hundred meters. And temperature out there is like 1.7, 1.8 degrees Celsius. So it's very cold and dark. And we can see all this because we have very powerful lights that illuminate this incredible place. We come into another sponge field with different species of sponges. There's a little crab down there. Oh, more of that orange sponge. Mark that, the orange sponge.
We just made up the points anyway, so I don't think that was it.
How long have?
Just look at the interesting stuff as we go.
Yes. What distance have we covered?
So we're kind of close to a marker.
Okay. Okay, I see it on the other side.
Yeah.
This is about 40 meters from that.
Great. We're an octopus now. Huh? We'll start seeing more and more of them. Here's another of those globular sponges that we saw before. Acabamos de ver ahora un pulpo en esta sección y esa esponja globular que hay que estudiarla. Podría ser una especie ya conocida o una especie nueva, al igual que esta amarilla que estamos viendo ahorita, con una vista muy hermosa desde arriba. It was a beautiful view of that orange sponge, yellow sponge. You could see the oscula, that's where the water comes out of the sponge.
So it just took some time, but yeah, it did make sense. Okay, perfect.
What's this?
Right there in the middle of the screen is a black coral, coral negro. Acabamos de pasar. Vemos varios peces. Thank you to everyone that's tuned in and sending questions and comments. We really appreciate that. And we're here. We can tell you what we're looking at in English or Spanish and maybe even Portuguese. Let me check if our colleagues are around. Not right now, but we have also people from Portugal and Brazil in our team. Yeah, there are people asking about the sea urchins. Yeah, we've seen several species of sea urchins, from very small ones to some large sea urchins we've seen previously. We'll probably come across more. And thank you to all the people sending nice messages and appreciations of this work. This is a unique opportunity for Costa Rica and for the planet to have access to this incredible vessel, the research vessel Falcor 2 of the Schmidt Ocean Institute. This vessel with its ROV, the remotely operated vehicle, allows us to go into these deep waters and just spend hours looking at these wonderful creatures like this sponge coming up, collecting these specimens and giving us a chance for the scientists to study the large animals, the small animals, and all the microbes that lives here inside, on the rocks, in the sediments, and inside other animals. So we have a very diverse team from many institutions around the world looking at different aspects of this region. Okay. Bueno, end of transit. Come to the end of this transit. Now we get ready, turn around, and go in another direction. Going up the hill.
See, it's just to come straight up the hill or go back. It's kind of along, put another marker on the line that we're at before.
Great.
You go back to the line and then travel on the other side and then up to marker 3.
Okay, perfect.
Yeah?
Perfect.
Okay.
That's good.
Did we finish the transit?
Yes.
Yeah.
Okay, you got it, okay.
Yeah.
You guys are in communications.
We're seeing cool stuff. Things we don't have. We're seeing the camera at the other side. See what's coming up.
Let us know when you're ready.
Yeah. Okay. Yeah, we're ready. We got a bit closer. Move us
There we go.
more of the line design or specific line.
Just like,
Plus, you know, we're just
oh, I see.
Yeah, so I would move the vessel a little bit closer to us because we're quite far away from it just now. But also move back towards the line because that's the direction we're heading. So maybe move it to where it's more of an area.
Great job. You set the waypoint where you started.
Let us know when you're ready.
Okay, I'll just get this set in a nice
Mhm.
place and we'll head off.
Yeah, we're getting ready to continue, and meanwhile we can appreciate these beautiful organisms here. Estamos alistando para el próximo transecto y mientras tanto vamos a disfrutar de estas maravillas que estamos viendo, esponjas, octocorales, y vemos ahí estrellas de diferentes tipos.
I'll hit start.
Okay.
You are, yeah.
Start.
Okay, starting.
So we're starting now the transit and we're moving now in another direction. We're still going up the outcrop. An outcrop is a rocky area that came up from bottom of the ocean in this case, and lots of organisms just attached to these rocks. Here we see a large sea star that we haven't seen before. And here comes another species of sea star. These sea stars. ¿Tú ves algo?
Sí.
Bueno, aquí seguimos con el segundo transecto que vamos a hacer ahora en la tarde, ya en la parte rocosa. En la mañana hicimos en las llanuras abisales, que es esos fondos lodosos. Y aquí vemos ya todo otro grupo de organismos que estamos en esta escena. Vemos esponjas, un pepino de mar, muy diferente a los que vemos en aguas poco profundas cuando vamos a bucear. Y bueno, hay secciones como esta en que no se ve mucha cosa, pero si nos detenemos y ponemos el zoom de las cámaras, nos damos cuenta que hay muchísimos organismos viviendo ahí. Vemos otra estrella de mar, una esponja que parece una copa de champán.
You gotta wonder how old the octocoral is. It looks huge compared to everything else. Must be sitting in a really good place.
Fine real estate.
Yeah. Seamounts apparently concentrate currents, and the currents are, of course, what carry food for filter feeders. And if you land right where the currents are peak, you got dinner flown in all the time.
This fish.
You still doing our transects?
Yeah, we're on number 2.
Okay.
I'm just standing in for a few minutes for Jorge. Pilot, as you're driving along, can you feel a difference in the current when we're going over an area with a lot of filter feeders?
Yeah, well, depending on the head of the ROV, sometimes if you're pushing that broadside on, you
Yeah,
can definitely feel it affect you more.
it must just come wailing over here. Yeah, beautiful stuff coming up. We think of the deep sea as being still, but there can be some phenomenal currents with an ROV.
I'll make sure.
There's that pretty yellow sponge again. All right. I'm just going along.
Yeah, we see those yellow organisms. Those looks like crinoids, which is a group of echinoderms, same group as the sea stars, brittle stars, sea urchins, and impressive diversity of octocorals, sponges. As you can see, from very small to large sponges. Aquí seguimos en el transecto y vemos la gran diversidad de octocorales, unos camarones rojos que nadan frente a las cámaras de vez en cuando, una diversidad de esponjas grande. Y mientras más subimos en este promontorio rocoso, más organismos como estos vamos a ver, que estos son típicos ya de zonas duras y no como en la mañana que era un fondo blando. Here are our octopus that we're looking for. There's the female octopus that are incubating their eggs That's what we've been. The mission of this expedition, main mission, is to study this octopus. There are probably 4 species, probably new. And fortunately, we had Janet Boyd here, which is the world expert on this group. And later on today, we'll try to collect some of the specimens, which we have to do because that's the only way that we can study them in detail, all their morphology, and do genetic analysis and be sure of what they are. And the importance of doing that is that we need to see if the species are found only here or in many different areas. That makes a big difference in terms of conservation. And that's why we collect these organisms and study them in detail, so you can see how exclusive they are from this region or not. And from our previous studies, we find that many of the organisms are found only in this part of the world. So that means that if they're extinguished for some reason, that's it. They're not going to come from some other place. Que seguimos con el transecto y mientras más subimos en la roca, más organismos de estos que viven sobre el fondo vamos a estar viendo. Ya vimos hace unos segundos unos pulpos. Ahí vemos una grieta donde están las hembras incubando. Esa es la razón principal de esta misión, estudiar estos pulpos. Y aquí ya empezamos a ver unas hermosas esponjas que son canastas de Venus y más pulpos. Y vamos a seguir viendo cada vez más pulpos conforme subimos este montículo, que tiene una característica muy especial, y es que sale un agua relativamente caliente. Y ahí es donde encontramos los pulpos y que ya están ahí cuidando sus huevos. So we see more and more the octopus, and we find them here because this outcrop is very special because there's warm water coming out from below the rocks, and we call it warm water. The warmest that we measure is 15 degrees Celsius, which is not very warm, but compared to the ambient temperature there, which is 1.7, 15 is warm. And this octopus come out here to incubate their eggs. One of the things we want to find out if it's because of the clean rocks, the water is cleaning, and the octopus need clean rock to lay their eggs, or because of the warmest water is helping them develop the eggs. So it's topics that we're investigating. As you will notice, there's more and more organisms as we go up.
That's us at the end of the second one. It's us.
Okay. And that's the end of this transect, so we now get ready to do our third transect and then move on to the very dense octopus field to do some collections.
A marker like 100 meters this side of the world somewhere. Where would you go? Would it go down there out there outside of that? You cannot let marker move it.
Mi mamá, la de Facebook.
Ah, sí.
Sí, desde la
¿A dónde lo estás viendo?
Esa es la que está poniendo ahí.
Ah, ya. Esa es su mamá. Enviamos un saludo a doña Lucrecia. Aquí está su hijo acompañándonos, Sergio. Buen amigo aquí, colega de esta expedición extraordinaria que estamos haciendo las aguas profundas de Costa Rica.
Hola, un saludo a todos los que nos están escuchando, que se están conectando desde Costa Rica y bueno, especialmente a mi madre. Un gran saludo y bueno, yo sé que también es muy fanática de la ciencia y la exploración, así que bueno, acá vamos a seguir y con la compañía y la narración de don Jorge Cortés, el experto mayor acá en Costa Rica.
Gracias.
We're ready to start the next one. if
Ready?
you want.
Okay.
Yeah.
Start.
Okay, start.
Bueno, comenzamos el tercer transecto que vamos a hacer esta tarde y como explicaba es una línea que seguimos 100 metros a 0.1 metros por segundo a 1 metro sobre el fondo y llevamos esos rayos láser para poder tener una idea de la distancia, del tamaño que estamos viendo las cosas. Esos son 10 centímetros entre los láser. Y así podemos saber que estamos viendo una banda de más o menos 2 metros de ancho que nos permite cuantificar lo que hay. So we started our third transect of this afternoon here at 3,000 meters of the Pacific coast of Costa Rica. And we're moving on straight line, that's what a transect is, and determined speed, which is 0.1 meters per second, 1 meter above the ground. And with the laser beams that are 10 centimeters apart, and this way we can quantify, we can look at this image that you're looking at and count how many Venus baskets, how many octopus, how many octocorals, and that allows us to compare with other regions right around here or other parts of the world that use the same methodology. Te mando saludos. Te mando saludos.
Ah, sí, gracias. Yo soy súper fan de usted. Sí.
Qué bonito, gracias.
Sí, está bonito. Como que los domingos la gente está más tranquila en la casa.
¿Verdad? Aquí vemos una marca, la marca D. Esas marcas se dejaron hace casi 10 años y de un estudio previo, y eso nos ha permitido volver a los mismos sitios y poder comparar cómo cambian. Y lo que es sorprendente es que en 10 años hay pocos cambios, hay poco colonización en esas marcas, lo que significa que si dañamos esto va a tomar décadas, si no siglos, que se recupere. Por eso es la importancia de cuidar estos fondos. We just passed a marker, already some of you saw that, marker D, that was set down almost 10 years ago. And what's amazing is how clean those markers are. And how clean those markers are. And what's amazing of that is that in 10 years, we get just a little bit of organisms growing on those markers or in the area, that means that if we damage this place for some reason, it will take decades and probably centuries to recover. So that's why we have to take very good care of the deep sea. Organisms grow very slow, reproduce at old age, and live for very long periods. So they're very sensitive to changes and very hard to recover once they've been impacted. Well, it's a question if the octopus are moving into the pots we set out yesterday. We'll check them on, see if they like the pots to move in, and we're gonna be dropping some more today to see if they are interested in that. And the idea of that is, these are beautiful octocorals there, the orange organisms, soft corals, and a brittle star. The more we advance, the more interesting this gets. But as I was saying, the idea of the pots is to see if the octopus colonize them because they are clean. That means that they're colonizing these rocks where the fluids are coming out because they're clean. Or if there's no difference, then probably they're colonizing this area with the fluids because not only they're clean, but because it's warmer. So that's part of these studies we're carrying out here.
Okay. There's a question about if the octopus have any predators around here.
I was going to ask Janet to answer that.
Question.
Yeah, here.
Why are the females postured as they are? I don't know. I mean, they have only a certain amount of space and they have to fit in there. And I wrote a paper once talking about the behavior in this genus, and I think they prefer to sit like under a teacup with their oral suckered surface on the bottom and their arms curled around over their back and running through their eggs. But of course, there's no teacup-shaped spot here, although there must be like a ton of spots that are acceptable. So they're just doing what they can. And those of you who are aficionados of octopus nurseries have probably seen the video from Nautilus Live, like you mentioned it. They're in a different position. Now, clearly, to me, these are different species, but I think they're just being adaptable. And unknown explorer Lisa, thank you so much. You made my day.
And, that's the other question.
Do any deep sea predators hunt these here? Well, what we're seeing are these tusked eel and shrimps, and they seem to both prey on the eggs. Now, it's notable to me that on the one face of the outcrop, you get the eels, on the other one, you get the shrimp. You wouldn't think they'd partition habitat like that, but, you know, that's biodiversity. I don't see anything here that would be a threat to a grown-up octopus, but one of the males who was walking around was missing the tips of his two left arms. And I guess when you're an octopod geek, you notice those things.
Thank you, Janet. Another question for you. What type of octopus are there? Or this?
Oh, these are octopuses of the genus Moose octopus. which used to be the genus bent octopus, but because of taxonomic rules, the name had to change. Of course, there's no common name because, you know, they don't interact with people very often. I think the shallowest one is, 150 meters depth? And, you know, scientists interact with these
I'm
ones.
saying, like, if we're going around like 10 kilometers.
Actually, the shallowest one is about 15 meters, and it's an unpublished work. Maybe it's published. But they lack an egg sac. That's the big deal that defines the genus, and they tend to have smooth skin, double sucker rows, which they're conveniently showing you just now. I think when they extend their arms into the water column like that, they're trying to increase their chemoreception ability because the suckers have chemoreceptors, and they may be sensing an odor trail that's invisible to us. Yes, ma'am?
No, sorry.
Oh.
Está aquí con nosotros la doctora Janet Boyd, que es la autoridad mundial en pulpos. Un gran honor tenerla aquí con nosotros. Y estaba explicando sobre esos pulpos. Son especies de aguas profundas, muy poco conocidos. Y ahora hace un rato vimos una hembra que no estaba enrollada, sino más bien abierta. Y eso probablemente están detectando compuestos químicos en el agua. Ellos tienen receptores químicos como en los succionadores, casi como decir una nariz nuestra, como nosotros podemos seguir un olor, ellos siguen un olor entre paréntesis en el agua. Y acabamos de pasar un octocoral grandísimo, debe ser varios metros de altura y diámetro. So we're seeing a lot of interesting creatures, and we just went by this huge octocoral.
Oh, yeah, that's all published.
Shark could have a field day here, but he'd bump his nose trying to get these octopuses. I think that's why they're hanging out in the premises in part.
Well, it's an interesting question there. Then in these places where fluids are venting, there's a lot of bacteria, but the octopus are probably not feeding or benefiting from this bacteria. It would be more the removal of the sediments or the temperature. But we have a large group of people working with the microbes. Also, they're taking rocks, looking at the bacteria living on the rocks, in the sediments, and not only what's there, but what are they doing. And we're also collecting samples for a colleague at the University of Costa Rica, Keylor Rojas, that's studying the fungi, plus samples for many, many different people looking at different aspects. This is this is an opportunity, a very rare opportunity. It might be years before we come back into these deep regions, so we collect as much as possible when we have the opportunity. Collect images, collects all sorts of samples. Okay. Okay, now we move up the marker 3.
Marker 3.
And then let's see the instructions. Mhm. That's very good. Thank you. Yes.
ROV approach to decode.
Yeah, we've got an updated waypoint bridge.
Updated waypoint. Excellent, thank you.
Make notes that were moved from marker 3. We're seeing looks like pillar lavas covered with large octopus. We're gonna set the pots
Okay.
in this marker 3. That's the idea.
All right. Okay, then we collect
Mm-hmm.
egg masses. When does she propose to recover the milk crate?
Let me see. I don't see it here.
I ask because I think there's an email attached underneath the group.
We are seeing several urchins here. This view, as we move into one of our study sites. So we're gonna move. We call it marker 3, waypoint 3, and just up in the rocks. And we're gonna move there and try to collect some of the octopus that we are studying. Again, thank you for your messages. It's amazing. There's a message from Brenda Jordan from Wyoming, quite a way away from where we are right now, and appreciate that. It's a beautiful state. That's one of those large orange sponges. So we'll get back to them later on. There we see another of our markers. That's marker F. As I mentioned, it's been here for almost 10 years. We're getting a message from many different places, United States, Costa Rica, and some interesting questions about the chemical composition of the fluids and bacteria, which I'll transmit to our microbiologists once they come up. They were around earlier. I'll let them know about these questions.
You can see that it looks pretty steep out there. And in fact, you can see here, we have a little representation of ROV SuBastian on the model of the outcrop here. It is even steeper above us. So quite interesting terrain up there, I'm sure. It is. It's scooting along. Yeah. You can show this barrel back it out. Yeah, Jimbo. Many thanks to Jimbo for getting some live telemetry on our model. Yeah, I'll put it up full screen. You can see that little ROV just scooting along. All right, back to the camera.
Almost there.
Okay. I talked to Beth, and she agreed that we could move that. Where was it? Oh, she forgot it. We moved the milk crate recovery here.
Okay.
Maybe with Octopus, so we're on the pilots.
Yeah, so it would be
Yeah.
probably easier to handle.
Yeah, and that would.
There it is. We're
All right.
gonna pick up that crate
Well, first, we're gonna get the deployments I have in the large bio box that's closest out.
Yeah, that's the first thing we need to do.
Yeah.
And so
We'll want them out, are we gonna put them down somewhere here or?
maybe
If you watched our dive, our last dive, we reached this point and noticed that the milk crate was still there. So we're going to try and grab that, I believe. They're talking about it over there. And then we also dropped off some projects here.
there's another marker.
That's a W.
It's, okay, oh, that K. We can leave,
W.
yeah, we can leave 2 here.
That's right, there's 2 by 4 at R,
Mm-hmm.
so we might as well put some here at W.
Let's do that. And
Anywhere in particular or just?
where they will settle very well, that they don't roll down. And it will be a pot and a tube.
Yeah, or on the tube.
Mm-hmm.
Look at that.
Yeah, here they're combined. So.
She's got repositioning, the deployments we made yesterday and mine down at J and K. And this is where that was. So we're gonna move that up here to B. And that'll free up, I think underneath these, there'll be females attached with eggs. And recovering this will be lethal. So we might as well just, one collection. Oh, there's 2 inside there.
I'm thinking a landing spot. I'm not gonna disturb anyone. Oh,
Yeah.
In my mind, I was imagining that we're gonna pick up the other one, because that's where your traps and stuff are.
Yeah.
Okay.
We're just reconnoitering right now, but we figured there's traps down there, so maybe we should put traps up here. And you
Oh, for
did,
deploying the ones you already have?
Yeah.
Yeah, because we need to empty the.
Yeah.
and you did have that as cheap, so I just moved it up to be
Okay.
recovering.
Yeah, yeah, yeah.
You're not forgetting things.
No, I was just thinking, I didn't put in here to be specific about which milk crate to pick up, so, it doesn't matter, but if it made sense to pick up the other one, since that's where we're gonna try to redeploy the traps that are already out. But either one, I think it's fine, doesn't really matter.
Okay.
We hope to get a photo.
yeah.
Just one,
That's okay.
on this dive,
Yeah,
we're only
maybe
picking up
I'll
one.
move my position a little bit.
Yeah, yeah,
We're gonna do
eventually
like
we're
our
gonna-
porch out here without disturbing someone.
So yeah, if you're still positioning yourself, no worries. I thought we were just
Either
chilling
way.
until we knew what we
Great.
were doing.
I can definitely do that
Sure,
for
Jill's mom.
which ones where.
Oh, no, oh, nice, okay.
And she said, he went on.
Oh, so cute.
Place was all.
I was
I want some
that,
shots, yeah.
like on Sunday,
Yeah, whenever we can get them.
laser at this one's like Discovery Channel.
Oh, nice, all right, so you're Discovery Channel. please.
Well, that's the idea we have is pick up the crate and collect the octopus. In these close-ups that we're looking at, you could see sea pans. It's these orange organisms attached to the rocks. But these ones are very different from what we usually see that are buried in the sand. They have their foot in the sand and the polyps up in the water column. Aquí estamos viendo detalle plumas de mar, que lo interesante de esto es que están pegadas a la roca. Normalmente lo que las vemos en aguas poco profundo es un pie que está enterrado en la arena y los pólipos afuera.
Or what the SOI team is.
will be interested in putting the pots in a place where there's venting, or
One of them, yeah.
yeah, a pair in the place where there's venting, and another pair where there's no venting.
Or just where they get warm.
Uh-huh. So let's see.
I think we're stable.
Okay, we're changing it.
We're still waiting to get fault and choose from somewhere here. Let's see, yeah. One thermal, one showing that and one not showing that. This is a black hole.
No, no.
All right, we just got a little bit of a handover going. And the scientists are currently discussing what we're going to be doing up here.
Chill. Welcome.
Okay, so.
So the idea now, we're gonna go into the bio boxes where there's the pot and the tube, and we're gonna put them, I would say, where,
Put it down.
where there's a lot of worm fluid coming out.
Which
And-
box are they at?
In the bio boxes,
On starboard?
I think there are one, bio box one. No, sorry.
We're taking a concentrate. Then we'll collect the milk crate, which has 2 octopus in it.
So that's like a 2-in-1.
It may be, but the milk crate will not be secured during the ascent. So we'll have to hope that they want to come up,
There are
or upon a condition.
bio boxes 2 and 3. So and it's a pot and a
They bought the,
how do you call that in English? How do you call that type of tube in English? We call it an elbow.
Elbow, yeah.
Elbow, that's what we call it.
I thought that give 2 a little more privacy.
Yeah,
And-
but that's the word you use in English for
Right.
that type of tubing.
Other, they're short, sold straight lengths and 10 foot.
Right, oh, yeah. No, this looks better.
So there's actually 4 pots in here.
yeah, where would you like to place it?
Yeah, more towards close to this area.
With the,
Impact of
If,
that
if,
shimmer.
if it's too far, now we're gonna set this tubing elbow and a ceramic pot, and the idea is see if the octopus are interested in in colonizing and using these to lay their eggs.
Yeah, I think so. Pot, right?
Yeah.
Sure.
So it'll be one-on-one.
So both
Thank
of
you.
these, Jorge, will go.
Yeah, let me check. This doesn't seem like to be like a very active site.
Yeah, we were just gonna set them here and then grab the other arm and just set 'em
Well,
there.
is that we went two in the very active side and two in the not very active side.
I think we'll have more freedom though once the milk crate's gone.
So what we could do is just, yeah, leave these two over here and get the milk crate and put two where the milk crate is.
Right.
So sorry.
Yeah, so no, leave them there. Leave them there for now, and there's the milk crate.
It's 2 and 4.
Oh, over here, yeah.
Yeah.
So you want that,
So that milk crate.
it's gonna go back on the front porch.
Yeah.
Okay.
Now that
And
milk
once-
crate has 2 octopus inside it.
Okay.
So we'll see if we can collect those octopus and put them on.
For anyone just joining us, you are watching live footage from the remotely operated vehicle SuBastian. We're broadcasting from research vessel Falcor 2, owned and operated by Schmidt Ocean Institute. And we are currently exploring about just shy of 3,000 meters below the surface of the sea at Dorado Outcrop. Currently, our science lead is Jorge. And then we've got Adam and Chill on the ROV controls here. And my name is Alex, and I'm currently serving as the multimedia technician on this expedition.
Now what we're gonna do is try to recover milk crate with some instruments that were left here years ago, and at the same time try to collect 2 octopus, hopefully a male and a female, and some eggs also. And as I mentioned, this is part of the study that we're doing. To see what the species are and what are they doing here.
Yep, so there were some questions earlier about whether we'd be collecting today. And it sounds like we're going to attempt that. Again, anyone who watched the dive last night. Our pilots were quite skillful in being able to collect these animals, but still understand that it can be sad to some viewers.
Hello, Les. Yes, we collected those sea pens yesterday. We'll probably collect some more. Unfortunately, when they were preserved, the polyps just fall off. So we have beautiful images and this mixture of the main branch plus the polyps. So yes, we have them.
So if you grab the box, like tipping it to track the octopus in there, are these other sensors secure in there? Are they gonna fall? There's zip tied in there. I would assume so, right? There's zip tied in. Oh, I guess. That was definitely zip tied.
Zip tie, I think, is too, yeah.
Before you try that, I suggest you get the shelters out of the bottle box, so that's available in case somebody wants to jump into it.
Sure. Okay.
Just a thought, there's no way we could put those two octopus into the forward, this bottle box right here, because I think he might just be able to go like this, where this one's quite good further back. He might have better success just dumping in there and closing the lid.
I believe Beth has plans to put rocks in there,
Okay.
Yes.
and coaching scientist rank.
Just, not clear for you to coach out. Oh, you're already.
Yeah, I'm halfway out, so I was just gonna see, let's pick out the view and then I'll porch out what you have in there. Oh, I guess you have to porch out to get the, sorry, you have to get the experience.
Janet, we have a question. The second non-brooding octopus we collected yesterday, that was a male. Were we able to confirm that once it was up top?
Absolutely.
All right.
A German. And what was strange, that male was roughly twice as big as the female,
Okay.
which, you know, if I have one and the other, well, obviously it's sexual size dimorphism.
Mm-hmm.
To refute that, to test whether or not that's right,
Oh, yeah.
I keep two of one of the sexes.
Right.
Which is why we need to collect more.
We're going to collect some more to see if that's a thing.
Just take a look at some of these when we have a broader angle view.
Mm-hmm.
And the males with their arms, you know, showing their web, really look big.
Yeah. So yeah, if you didn't quite catch that, we did collect both a female and a male, and it was noted that the male was notably larger than the female, so that seems to imply some sort of sexual dimorphism. But of course, with just two specimens, we can't say that. So, in collecting a couple more here, we can hopefully reinforce that a bit.
seems to be on the dorsal rather than the ventral surface.
No, I think it's. What's the interesting part of it? The males have a distinct honor feature, and so you know the difference between the male and female. But I got, assuming these are all females that are breeding eggs, so it's a male in that position. Yeah, it's like this little, see that little like white, sort of shape.
Are you able to grab a center porch or anything like a better view?
Yeah, we
Yeah,
can
it'd be
zoom
nice to see when it wrote it, and I doubt it, but it could be there's a male underneath. And I just don't understand. These are not necessarily all brooding. I think we should know that if that's happening. Because something really interesting I saw yesterday when we did dissect a bit of the gut is it looked like there were pieces in there. I think they might just be living there.
It's,
ROV Bridge.
Jeff doesn't remember these very well.
Hey there, how would you feel about us adjusting the ships heading to port by about 10 degrees?
Yeah, that'd be fine. I'll just, put the waypoint about there and,
Okay, I'll move on to that waypoint and then do the,
after we
heading check.
get it up. Thanks, Steve. My final kind of hard task is trying to do that tipping. I feel like if it just can move independently of my arm, I think I'm better. I don't know. Worst case scenario, though, you can set it on the porch somewhere, just
I
grab
was
it.
not super painful, so I don't wanna interrupt and I'll wait till this is all.
I think we looked at it, it's everything zip tied and then.
Hi. Yep. Catherine and Jeff's 9-year-old has been over there.
All right, on the far side,
Been here since 2014.
on the port side, you got an octopus on the bottom. Could you take her over the large file box?
Right before the move for the, done that now. Okay, everything in there should be zip tied, so it should be fine, it shouldn't fall out. Yeah. Yeah, okay, I'm good with that.
All right, so we notice there's an octopus in this crate. They're gonna just try and use this as an opportunity to easily collect this specimen. Although it is an octopus, and from yesterday we know it will be anything but easy. Yeah, it turned out there's one on the outside of this crate, so the pilots are just gonna see if it'll drop off into the box. Oh, your mic's off, Janet.
Thank you. It's smarty pants. Pilot just turned on the mic for me. We're doing this recovery of this instrument, and there's an octopus who's colonized it. And we're not going to leave the box because the octopus is on it, so we're trying to recover the octopus and the instrument.
Yeah, okay. Or even if I came down, then you could, come down.
Dang, that's a lot of eggs. All right, here's our.
Okay, we'll hold it a little pressure against you.
9 years. Oh. Then, if we're not successful in getting an octopus to come, you can also put the milk crate on the basket with that side facing out, so at least it has the opportunity to come off if it wants to. And then try to actually get an octopus the way we did yesterday.
Well, the other thing is, there's two octopus in there.
Yeah, there's 2 octopuses.
Yeah, I was gonna say, we flip upside down and strap it down, and then they may or may not go.
That would be problematic for the instruments inside to incorporate.
We looked closely, they looked all them strapped down at the same time.
Yeah.
And I think even if they did fall out, they would strap on between the grating and the offset down. So we've got a strap where we're gonna let you over something.
Currently, just a discussion of how best to approach the octopus on the box here.
So, yeah, my concern is that, on the left side, if it gets hit, it will crack, and we'll lose the sample that's out of it. But, it looks to be flush or lower, so I would be able to do that. Yeah, it's right there.
What about those handles? Would they be able to escape on those handles? Upside down?
Oh, they'll definitely escape if they want to. Yeah, so I would
Maybe it's not worth it.
No, I think we can make it work, because also the bridles there give it a little bit of padding, so that it probably won't crack that top part.
What about this? We try to scrape this guy off, if that's successful, then don't worry about it, it's like a box right side up.
Yeah, exactly. We've got the 2 inside. My point is, these aren't the only ways that we will try to collect them.
Yeah.
We've got one on the outside is troublesome, then just put the milk crate where it was, let it decide if it wants to come back or not. And then try other ways of collecting them.
So I'm now trying to collect this, put this octopus into the box. We can see it, a female with the eggs, and try to collect the eggs too.
Get some babies coming up now. What was your decision on this? I think it was I doubt it's.
Yeah, that's.
And the ones inside. Yeah, I like that. That's a good idea. They're really
Yeah, so that's not worry about that. Do you wanna just set it down and regrip it so you can turn that octopus so she's facing out?
Yeah, that's what I'm suggesting, turn it 180 degrees, so the octopus is facing outward, and then have the choice of whether it wants to come back with us. And then that allows us to still do the nets
Yeah, we thought about slurping them, but uh, the octopus are larger than the diameter of the tube, so it will probably damage it very much. Uh-huh.
What's that for, right?
It's a probe, temperature probe.
Is that coming back with us too?
Yes, yes, yes. I guess because I wasn't here when they said that. That's what it looks like. There's something else there.
I can also commit on the ramp, do like
Great. Yes.
yeah, oh no. Cause I mean, I guess you could. Did you want to try to shake that off into a bio box? Did you want to try to shake the octopus off into a bio box? Okay, so which bio box?
The proximal, the closer starboard one.
This one?
This one. The one you took the deployment center.
So the only thing is that this one's divided in two, so if you go in here, that. you
So
go
again,
over-
perhaps an opportunistic collection here.
Are you good with that?
The main system, we put one here, it's gonna close, and we cannot open it again, so it'll come out. So
Yeah.
we'll only have one. So what we could do is put one in there and put the napkin on top.
That's okay.
Let's see.
So they've got the octopus on this instrument, and they're just going to try and kind of jiggle it off into the collection box, the bio box. Excuse me.
But I mean, what we could do is, we could put the whole tube in there and close the bob off on it. But it's attached. There you go. Yeah, it,
Put the net on top, hold it down, put the other one in.
yeah, but it's with the rope. So it locks in that place. Well, it'll close enough that he would be able to get out.
Right,
but-
that's what we need.
Yeah, it doesn't matter. You happy with that?
I'm happy.
I think we just close it right on there. Okay. You happy?
Yep. So pilots have elected to just put the instrument in the bio box and just close the lid on the cable there.
So one's still attached to the front side of the porch. Could you get a net and see if you can bring her up? See how good your elbow is.
What's that, sir?
This net over here. Before we try that, though, should we just-
All right, and this is they were discussing using the net, which if you tuned in for our last dive, is how our pilots were able to collect the octopus previously.
Oh, yeah, sure. Go for it. Start sticking in there. That's right.
They've noticed that there's actually an octopus clinging to the front of this other bio box, clinging to the ROV itself.
Okay, so check it out.
That's.
speed bridge. Hi there, can I bring the ship's head around another 20 degrees to port?
20 degrees to port, sure.
Copy, thank you.
No, this would pull the waypoint above there too, that was there.
Okay, I'll drop back and then I'll bring the head around. Thanks.
Great, thanks.
Mm-hmm. Yeah, the voice you hear every once in a while, more over radio. That's the bridge and our pilots coordinating the movements of the ship. All right, well, it's probably a good opportunity to do a little reintroduction for anyone that might have joined us recently. Noticed our views have increased a bit. So you were watching live footage from the remotely operated vehicle SuBastian, which is being operated, and we're streaming this from, research vessel Falcor 2, owned and operated by Schmidt Ocean Institute. We're currently just shy of 3,000 meters below the surface of the ocean at Dorado, excuse me, why did I say El Dorado? At Dorado Outcrop. And we're currently trying to collect another specimen or several more specimens of this octopus species.
Better than what I had. I got it back forever now. Trying to get more straight on with it.
So those of you who haven't been following the expedition so far, you can find more about this at schmidtocean.org. But the thing that you might notice in the screen here is that there are multiple octopus present. And some of them are in kind of this odd position where they're upside down with their arms spread out. So what's been found and what's interesting about Dorado Outcrop is that it is an instance of an octopus garden or a nursery of sorts. These octopus that are upside down are actually brooding eggs. There's some males wandering around as well, which don't assume that sort of brooding position. And scientists want to learn more about these animals, why exactly they're doing this here, about the species in general. And so all sorts of samples are being collected on the outcrop, around the outcrop, and of the organisms here. Janet, we were able to confirm that these are a moose octopus. Yes. Okay. So as far as the species, I'm still unknown on the species, but it is a moose octopus. You do have the bungee, yeah.
Push back out and it's surgical right there. Yeah. And I'm sure the box will just kind of tip as I'm reaching in there, and I'll just try to pull it out underneath.
Well, and for anyone who missed it earlier, this crate has been down here for 9 years now. 9 years.
Okay, we recovered this crate and some octopus.
Everything else okay? More lids.
Thank you, guys.
So
Just, I think it's just bent. I think the mouth was on the left side, kind of popped off one that it was hooked. Right. And I didn't see it. But it's still there, so we're all right. Okay, so we need to reposition. For sure.
position the pots that in this area.
Currently discussing with the scientists about repositioning the pot and the pipe.
Stream.
And it doesn't matter their orientation. clearly the ponds are getting in. It is surprising there wasn't more colonization in that milk crate, but maybe corals are smart enough not to get in hot water.
Yeah, okay. Over here.
With these deployments, we are trying to put them at least where it's warm, whether in current flow or not. I think that's a little bit hard to predict. We're testing
Is this a shimmer
substrate
right
and temperature.
See the shimmer right there?
Right. That would be. Yeah, here.
There. Oh, yeah.
Yeah,
Lots of it.
I think this all is shimmering.
Yeah, yeah.
So if you just settle that down right there, then it'd be peachy. Perfecto.
Right. Okay. Let's just check the other two that were just put there. Make sure they're.
These guys over there?
Yeah. Make sure that they.
Are they away
Well,
from
maybe.
fluid flow?
Yeah, they're definitely, because you want those out of diffuse flow.
No, but there's no flow. That would be.
Two inflow, two out of flow.
Yeah. So maybe we can even push those a little bit farther up.
I think they're good. I
Right
mean,
there.
that flow is going straight up.
Mm-hmm.
We're having a test gill come in and check out the terracotta pot, BB. Or maybe it's going for the BBC. They're totally aware of something is going on that's different. That to me, to them means food.
Yeah, look at the shrimp going in.
Shrimp are looking for food too.
Mm-hmm.
There's those eggs right there. Can we hold that shot for like 10 seconds? Of course. Thank you.
I think you can close it, too.
Yeah, I mean, if we can just get some long shots this dive if we have time. I'm always for it.
We have set up the experiment, the pot, a pot and a elbow in the where it's you can see the shimmering. That's the warm water coming up, and there are two others, a pot and a elbow set up in an area where there's no warm water coming out.
Get it around.
And before we move on, I need to see the other pot. Yeah, I miss the number or the letters. It's the letters. Maybe we can just see it from here.
4.
The number 4 and
2. Yeah, 4.
the pot
The
is
clay
2.
pot is 2 and the pike fitting is 1.
I think they're where it's cold,
I think they
and
were.
I think there'll be an encounter from the octopus, so that's all we can do. Well, that shrimp shot in the pot, shrimp in the pot,
Yeah.
that could have been a video highlight, depending on how you liked it.
Right.
With like
I can just make a new, the new owners already.
We've got the eggs. Lab samples, the laboratory's
right here from the venting area, we need to get a rock
Rob, do you know how to pick rocks to collect?
and
Sorry?
Do you know how
a
to
rock,
pick the rocks
and we
to collect,
put
the
them
letterbox one?
these
I don't,
boxes.
I'm not.
And you want it from the venting fluid?
Yeah, for the
Oh.
venting.
Okay,
But there's a
you
lot
would
of
think-
venting.
I think if you always
Where the
want
clay
to
pot
get
is?
them.
Right. Like right where the clay pot is in a couple of
Yeah,
rocks?
yeah. Yeah, one nice rock. And then we're gonna fire Niskin bubbles.
Should I ask Beth to come in?
Maybe, yeah. We'll do that too.
So when I did the deployments, I labeled the clay pots both to medium
Yeah.
and then
Yeah,
the,
me and one.
okay.
So
So,
the active side and the other one is up to and over 4.
so it's okay to have like duplicate entry then, other information, okay? So this one is, clay pot.
So we've done that. Up to and over 4.
This is the non-active.
Rock selection time?
Yes.
Okay.
I have few notes here, so this we're transcribing like that. So we're here.
Right, and this is where we picked up the Osmo sampler.
It was exactly there. And you could see once you get close, it's
Yep,
lots of shimmering.
okay. Can I control the camera for looking at, oh, no, just for looking at rocks. Yeah. So where, it was over here, right? Nope.
So the Podgy and one are in the active side. Okay, so I got one.
Also made a note that there are 2 octopus
Yeah,
You have a
so
preference,
I-
Gus?
They all look pretty good. I guess we need the right size. Is this going on equipment or firebox?
which might be
Going
That's right.
in the
Yeah.
forward bio
And then
box.
I've got one that's in.
Can you put on the lasers, please?
We'll see what's there.
if it comes up.
Yeah, but I've recorded them as samples, and so they're in here.
So we'll see.
Okay.
We'll see if it wants to be great.
Are they on? I just can't see them. There they are. Now I see them.
Then the other one's
Everything here is kind of tiny.
So she may actually be okay with all that stuff in the front. But the temperature was
What is worth our time? Sorry, Octopus, don't mean to shoot you with lasers.
Yeah, so we have to reposition traps yesterday.
We're not there yet. This is a different site.
Yeah,
Yeah. Okay. Before we pick a rock, can we get the high temperature probe so that we can poke around and see which ones are actually in the fluid? It's always deceiving, looking through the shimmering water. Okay. Oh, sorry. Where are you going, buddy? You gonna hide the rocks I want? I'm basically interested in getting at those rocks right underneath that octopus. We might want to let it figure out where it's going. All right. Where can I see the temperature data? Okay, thanks. So it seems the flow is coming from the right to the left, so, and closer to that PVC trap. So, okay. Maybe right where that shrimp is would be a good spot. Look at this new feature that we have. So in your picture-in-picture, we have a way for you to see the temperature readings that are coming from this high temperature probe. The moment you can see that they are not very warm. Let's go ahead and pull out of that little crack. And yeah, where is the flow coming from? It's basically coming from underneath that pot, maybe.
I'm seeing a shimmer right here. Don't know where it's coming from. Right in there is shimmering.
Yeah, give it a try. No, you made a good guess. Is it possible to penetrate any further down there? Or are you already hitting refusal?
Yeah, that's pushing the RV up.
Okay, great. So it seems like it's stabilizing out around 10.1-ish. So if we could record that, Rob, 10.3. All right, you can go ahead and stow that probe. Hello, fish. So these are all pretty small, where he was just measuring. Do you want to get a couple small ones, or do you want to get one big one?
Kind of torn. Can we get 2 medium ones?
Not sure. I mean, one thing we could do, I'm going to bring up a gun,
Yeah.
is maybe grab the scoop
Yeah.
and try to
Oh, that'd be nice.
scoop some of the small ones and then dump them in the buy box.
The rocks in
Yeah. Where is that dive plan piece of paper?
Yes.
Yes, thank you.
I think it's just the LG restrictions. You're hanging out.
Yeah.
The next guests are coming up after this?
Yeah. And then we'll probe some more and try to fire one of those squeezers.
I'll try.
Do you want to direct this, the rock sampling? Okay.
If you have to get back to work, I will.
No, it's fine.
Otherwise, I feel like we're experts.
Yeah, it's,
Oh, wow.
vertically,
Yeah, appreciate how far out that science camp can.
oh, the net.
Go
Scoop
forward.
net.
The scoop that's hanging on the breaks that day.
Yeah.
I think there's any more brine left in the Osprey.
Doesn't look like it. I sent a picture to Jeff. He didn't seem to think so. It's always misleading looking at the plastic that's separating the two reservoirs, but it doesn't look like there's much.
This is good to recover the harbor and we get an octopus.
Yeah.
It's part of the dive plan.
Oh, sorry, not that one. Do we have the, wasn't there like a metal one? No, it's just these bags.
But this one doesn't work too bad. It's got a metal edge, so you can scoop with it. Or maybe you just
Yeah, it's not good for the purpose of what we're getting the rocks for. I don't want them to have this dirty bag all over because we're gonna be sampling their microbiology. I thought it was just the metal scoop. That was my bad.
Okay.
Would you
Getting 2
agree?
medium ones?
Yeah.
Picking up
Yeah, so we can go ahead and put that back. I apologize for that. It was my bad for not knowing what we had in the basket.
yeah, who knows what's in that bag, right?
Yeah.
Almost the last in there.
Well, there's a lot of
But
human
instead of
DNA,
yeah.
I'm sure. And it was used to collect an octopus yesterday. Yeah, so for the audience at home wondering why we're so persnickety about this, we're interested in studying the microbes that live on the surface of these rocks exposed to low temperature fluids, and then how they might be different from microbes that live on rocks that are not exposed to low temperature fluids. We think that microbes perform really important functions in this environment. They might be producing new carbon for the food web. They might be involved in cycling nutrients and metals. So it's important to us to try to get as clean of a sample as possible. So we'll try to use the minute to go in there and grab the rocks. All right, so then, looking back where we were. Yeah, of course, that octopus went right to where we did that temperature measurement. But I think these rocks, if we want to maybe, how feasible is it for you to try to get the minute in there to try to grab in this area?
Those are pretty small.
They're pretty small. I don't know.
As long as you're happy with that, that's fine.
Yeah, I feel like we'd try it just because that's where the flow was, that kind of area and that kind of material. If we're not successful, then we'll try to get something bigger. But that's the stuff that looks like it has the most manganese oxide coating on it, which is the most strong indication of flow. Okay.
That's one by box one.
Yeah, buy box one. It looks like there's already. Is there already something in buy box one? What is it? Was it collected in the low-bait field? Okay. It's okay. We can try again.
What about right here? We're kind of where the shrimp is. There's a lot of shimmer. I know that's a little higher than where we measured. What do you think about that guy? And even maybe if I flip it, look at the other side of that rock, see if it has the material that you're looking for.
That's a good suggestion. I like that. Janet, it looks like one of the octopus is already starting to explore your PVC shelter.
I'll bet next week they will move in bag of baggage.
Potential sites for that showing one over there. This one over here, but octopus is step right in. Yeah.
So can we get a frame grab of this, please, Rob?
That suggests that most of the darker rocks
A couple of them.
are supposed to be
What you're looking for?
more manganese oxide than the greener rocks.
Yeah, so it's got some of that black manganese coating on the bottom. It's maybe not as thick as on the little rubbly pieces, but it seems like a good piece. Do we have, yeah, and it's maybe about 10 centimeters, so it's still on the small side. We might try to get one more and put it in the same position in the bio box. But yeah, let's go ahead and stow that in bio box 1. And yeah, in section B. 1B, please. Nice collection. Chill. It's for microbiology, yes, Rob. Thank you. Okay, let's try to get one more if we can. And now that actually we pulled that out, it looks like the shimmer is even more. You can see some old egg sac cement in there. Yeah. What do we? You want to try that one that was right above it? Yeah, that's not in the area of flow. I want to focus and get 2 rocks that were in the same fluid.
Sure. This one here?
Yeah, let's try that. Oh, there's a little fish. Oh, there's a little baby. Hi, baby octopus.
That was cool. Still pretty cool in here.
It's actually got 2 layers. It's got manganese and iron oxides on it. That's a great picture. If we could get a frame grab of that. And if you could hold it steady for just a second.
Sure.
Thank you. And, oops, sorry. Well, let's get a nice little, those are like some iron oxide mats. Those are beautiful. Okay. Chill, would you be able to rotate your manipulator 45 degrees, 90 degrees or so? I want to see if that rind, if that's a rind. Yeah, that looks nice. I think that's a good one. Okay. Thanks, Chill. We can go ahead and put that in the same spot, bio box 1. Settle down now. So that's also going in B. Yep. Okay. Okay, that's fine. I'm gonna do a little bit of switching stuff up so our audience can hold on for a moment. Yeah, so I wanna, we'll do the Niskins and then we need to do some more probing and the squeezers. And I would like to be here for the squeezer part. But, you wanna come back in?
No, no.
Okay.
We can, it's better if you stay
Okay.
here and tell them I want the
Yeah, yeah.
squeeze or whatever.
Okay.
Turn off the blower. That's the one that I turned off.
Oh, that's a nice shot. Can you take a screen grab?
Yes.
Oh, also cool shot. Lasers. So for anybody that's new to the feed, watching the dive stream with us, we're currently sitting on the Dorado Outcrop, which is a small outcrop of rock in the Pacific Ocean offshore Costa Rica, where low temperature hydrothermal fluids weep out of these basalt rock environments. Temperatures are just slightly elevated, about 12 degrees Celsius compared to background seawater, which is about 2 degrees Celsius. And we see that octopus seem to preferentially be brooding their eggs and inhabiting this low temperature fluid. And so we're here to study those octopus. We're also here to study the environment more broadly, the microbes that live in this ecosystem, the chemistry of the fluids. Yeah, they're doing some technical stuff. Yeah, so that's what we're here to study, type of samples we're collecting. Got a few more hours left on bottom for this dive. And for the audience members that are asking us about these octopus, you'll see that many of them are in what's sometimes called a defensive position with their tentacles facing outward. Some of these octopus may be brooding eggs, and so protecting those eggs, but it's possible that not all of them are doing that. That's one of the things we're here studying is what is their behavior. And doing some cycling of lights and things. So you'll see the lights go in and out while we're correcting some things. Got a couple fish coming by to visit. Looks like one of these we saw yesterday. I think I recognize that one. Yeah, it's the one we saw last night. You guys still testing stuff, or are you ready?
Yeah, let's, we'll just launch this from there.
Okay. Yeah, just let me know. So our next operation would be to stay right here and to collect 2 Niskin samples, one of the 5 liter and one of the 2 liter. And what do we have left?
2 through 5.
Okay, so let's do 2 and 5. I'll let you control the camera for it.
Well, we put the empty space.
Can I borrow your sheet there for a second?
Yeah.
Left one. Thanks. We did do, so we got this guy here, and this guy got this here. Seal assembly. Plate assembly first.
Yeah, 2 and 5. All right, there's number 2. The difference between these bottles is their size. The number 2 Niskin is a 5 liter volume, and the number 5 volume is just a 2 or 2.5 liter volume. So slightly different samples for slightly different purposes. All right, and then our next operation is to do some more, go back to where we were just doing that rock sampling, do the T-probe again, and then we're gonna try to fire some of these squeezer samples.
While we're talking about it, can you go back to the left? So we did our temperature probe there, but see that spot right there? There's way more shimmer there, and there is a bit of a gap for the squeezers. What do you?
Where are you, I'm sorry, where are you
Right
pointing?
in here, just to the left of the clay pot, that kind of hole there, there's a lot of shimmer there. If
You're
you're
right.
interested in that area?
Yeah, well, when we do the T-probe, I think we're gonna probe around.
Let me bump over that, because I don't want to probe a little bit.
Okay. Yeah, ideally, we'll take multiple squeezer samples here.
Okay. That's what I was saying before. I don't know if we can actually get, I'll watch your, yeah, I'll watch your program. Octopus going over the clay pots over there. That's a good spot. So, what's coming up?
Oops, wait.
And I'll push up just a little bit. There. What's coming up? Make it rotate over to the. Do you want to just hold and take the camera?
Hmm?
Do you want to slow it and take the camera?
Mm-hmm. All right, thanks for getting that picture-in-picture up for us, so everybody can follow along with the temperature measurements being made. Can you take some frame grabs, please, Rob? All right, 11. All right.
Sorry, I gotta reposition just.
Okay, that's fine. The max temp on that was 11 that I saw. Everybody wants to be right here.
Something to be position this.
Yeah, yeah. No, it's over here. You gotta get the worm over here. Well, it's good guidance. It's getting warmer.
Yeah, I can't seem to find the power stop.
Yeah, that's good. 10.8, not bad. Okay, and then if we could go ahead and reprobe the spot where we collected those rocks. I think just to the left of where you are, over where that green cement is behind the octopus. Okay, if you could hold it there. 6.7, 5.9, 6, 6.5, 6.6, 6.7, 2. Come on, come on. There we go. All right, 10.3. Okay, we can go ahead and stow the high T probe. And let's grab one of those squeezer samples.
That's the issue there.
Do you have a preference on which one we do or no?
Nope. Whichever one is least annoying to pick up.
Transferring this into another, and you get that out while you're grabbing.
Sure, yeah.
So the device we're picking up right now is called a squeezer. It's a spring-activated syringe sampler, which we're going to try to use to collect a water sample. Can you hold that? Oh yeah, let me go. Keep doing what you're doing. I'm just gonna zoom in on the bottom there. Or do you? No, never mind. You need my view. Just kidding.
Were you wanting to look at that tube on the bottom?
I do, yeah.
Go for it, this is a good time. If you want, I can try to straighten that.
No, it's fine. I can get what I need. Hopefully that, yeah, I think that'll still work. And just want to check that the bridle here isn't going to potentially get in the way of the spring.
Sorry? I'm trying to tilt this a bit better if you want. I think just open up just a touch. What? I think, yeah, I think so.
Nicely done. Okay, if we could, let's see, where's the bottom in relation to us?
He's covering our spot, isn't he?
Yeah, that's fine. We can go to the other one first anyway. We can go to the one that we probed most recently, down to your right. And we want to try to angle that little plastic tube as much as we can to be down where we think that flow is coming from.
What do you think?
Let's maybe try to come down a little bit. I think yeah. If we can get it like right in that spot, if possible. Yes.
Ready?
I will be in just a moment. I want to see the pin. Can you take a frame grab, please, Rob?
Yep.
Good?
Got it.
Okay, let's try to squeeze this. I love it. I love it. Okay, so hopefully we got a nice sample there. Say thank you to Tim D'Angelo for getting that piece of equipment prepped, and to Jeff Wheat for letting us borrow this equipment. Yeah, it's marker K and marker W. They're like right on top of each other. All right, we're gonna do another one, please, over, if we can. If our octopus friends will let us sneak in there. Nope, that one's got a little plug of sediment in the bottom of the tube. Okay, can you put that in the notes though, please, Rob?
Yep.
Nope. We'll see in just a second the numbers on the backside. Okay.
It's 5.
Number 5?
5, yeah.
Okay.
See the dip candle? So you show you the backside of it.
Oh yeah, there it is. Number 5. Thank you.
Seems to be in there decent right there. We got it. That's kind of what I need to do. I'll do it. No, I'll do it. Yeah. Okay. We got our little hole where we throw it.
Yeah, it looks like the octopus is vacated, so we can get in there. Can you get a couple frame grabs, please, Rob?
Yes.
Thanks. Yeah, this looks perfect.
I believe that's the tube down and the track behind that rock.
Yeah.
You're happy with that?
It's a good setup. Well, let's pull back out because I want to see the pin, please. Thank you. No, sorry. I meant the camera, not you. Apologies. All right, let's just hold for a few seconds. All right, and go ahead and try and fire. Nicely done.
I know. She's not really getting in there, but she is.
We're gonna try one more in that same spot.
Very well today.
Yeah, Tim did a good job.
Yes, yes. And then with the water, you, what do you do with it? You preserve it somehow, or?
Yeah, we'll sub-sample it for a variety of things. So we'll take some samples for chemistry for Jeff. We'll preserve some for potential single-cell sorting, for counting the number of cells, and then we'll filter the rest for DNA analysis. And those will be sent to Julie.
Tube, you can open the door. Huh? Or maybe do the tube just standing up on its own.
It's true, yeah. That is what it is. Yeah. Oh.
All right, number 3. Is that any good? Yep. My watch.
I'm trying to slow down this.
Yes. that the same area for this one?
Yes. See if you can get there before the octopus does.
Okay. Awesome.
You can get a screen grab of that, Rob.
We happy with that?
That might work. Seems like it's still not quite in the crack. Yeah.
Let's zoom in there.
Okay.
That reposition. Back there. Pretty happy?
I think so.
Okay, go for a pinch.
Hmm. All right, this one seems to be a misfire. Might be because we're grabbing towards the top. Not sure.
What trying to be loud? Okay, good try. Okay, yeah. Pushing us off there, Nick. Yeah, I know, the shoulder's pushed through.
But that's okay. The tube is actually all right. Yeah, go ahead and try a pinch again. Might be a lost cause. Okay, so this one's not gonna fire. That's all right. We'll go ahead and stow it. And we'll try that fourth one.
Maybe give you a bit more porch out now, maybe it was moved. Yeah.
Well, I didn't know if you were like wanting to make sure you got.
I want to make it,
this.
I want to just get it screwed out.
Okay, I'll let you, like, just tell me when you're ready, I'm right here.
Okay.
It's just so exciting.
Ready?
Okay, I'll need it.
Tube is also bent kind of sideways.
Yeah. Is it? It's at as far back as you can go.
I think it's close.
I'm looking down and it just.
Try one with the
Also seems that.
That's probably it.
Yeah. Okay. Yeah, there we go. That's a good shot right there. Zoom just a little bit back in so we can see that tube. There we go. Let's wait for this octopus to maybe get out of the way so I can see the tube when it fires. Or maybe the octopus wants to take it with it. Come on, buddy. Okay, screenshot, please, Rob. Okay, go ahead and try to squeeze. Another piston stuck. Okay, well, 50% success rate isn't bad. Thanks for doing that. Okay. Out of the sediment, they didn't find as much stuff? Okay, you want to do the repositioning stuff? Okay.
Let's see. Yeah, we have to reposition the microbial experiments, the traps, the little tubes. You know, the Gustavo here, so make sure we put them where he wants them.
So next up.
Yeah, well, we have to find the tubes that were put out yesterday. I think they're very close by. Let's zoom out.
They're close to this spot, so do you want to move off here and have a fly around and see if we can see them? Or?
I think they were, if I remember correctly, they were close to the basket. And the idea was that once the basket was removed, they would put them in there.
Okay.
So they must be, they should be around this or close to the other basket. So let's zoom out. Yeah,
Come off and we'll have a little look around.
Mm-hmm.
Going to spread the experiment?
Yeah.
All right.
There's the other ones. Something like the leaves.
So Gus will take over for this part. That's his experiment. You remember exactly where we were led?
I'm right next to marker R. Yeah, no, that's R right there? Oh, that's me, okay,
That's
yeah.
it.
So,
So the idea would be to pick those up,
and just spread them out a bit, but hopefully around
like where
the
other
most
basket was.
possible shimmering, you know, where they would fit,
Yeah,
yeah.
we can just pick them up and go back to where we just were.
Yeah,
Because there's a lot of.
there's a lot of shimmering there, exactly, yeah, so we could just take those over.
Picking them all up?
Yes, you may pick them all up. I think that if you can grab two, that would be, 3 and 2, that would be 2 trips. Not sure if it would be a good idea to do all 5, just because it'll be hard to navigate them landing without hitting an octopus, apparently.
Yeah, I mean,
Yeah.
we can grab multiple in the T4 at once,
That's fine with me, yeah.
and then just take
Right.
them over and we've got to do a couple of trips and do it.
Okay. Yeah, right. So, yeah, so these traps is just sterile metal and rocks, and they're going to hang out here for 6 months. Try to find them when we come back in December.
Still there.
With this experiment working, and then the experiment that we left in the nodule field, and at the rock field today, that's all that I would need to do compared to. Fingers crossed, I got 3 more potential deployments at new vending sites that I'm able to do.
Great.
Stay over there. We will just try and grab as much of the ropes in one grab as we can and see what we see how good we get it, and then we'll just go back and come back some more. We're happy. Yeah. Try and grab as many of the ropes in one sort of swoop.
So you gotta grab some with the other arm as
Yeah.
well.
Just stand by with that before we walk it at the side of the stream. Until we're done with this one. I know, no, you're fine there. Just keep it at the side of the screen, but lock it. You know how to lock it, so you go into the, just text it, lock it, or press that. And that means if you nudge the freeze button, it won't come unfrozen, you don't notice. Always good to do.
It's just
That happens.
colonization experiment transfer 1, and that's it. That should be enough for me to chase it. Transfer, yeah. They holding on?
That's amazing for sure.
Hello to all the viewers out there. My name is Dr. Gustavo Ramirez. I'm an assistant professor at the California State University, Los Angeles, and what we're seeing here is simply the transferring of a single colonization experiment left behind here yesterday by the ROV pilots while we figured out where was the optimal site for their longer-term placing. So these PVC cutouts, the small PVC cutouts, contain 5 types of sterile substrates that we think can be informative in regards to better understanding the microbiology of the fluids. So these initially sterile substrates are supposed to hang out at sites of water shimmering, and over 6 months of deployment, ideally collect microbes that come out of the shimmering fluids, that is, collect microbes on their surfaces that come out of the shimmering fluids rather than the background seawater. And again, these microbes have an incredible capacity to degrade or utilize a lot of the minerals that are found in these substrates. So the deployments are as follow. One of these traps contains 23-million-year-old basalt that is native basalt collected from this site 9 years ago. So I was lucky enough to get a hold of one of these rocks. I took it to my laboratory, sterilized it, and then crushed it and are deploying it back here. Pretty cool comparison to that one is going to be another set of PVC experiments here that contain 20 euro basalt from the place where this 23 million euro basalt was born 23 million years ago. So the western side of the East Pacific plate, East Pacific rise, I'm sorry. So we are on the eastern East Pacific rise here. That again is 23 million year old seafloor. And so I've got seafloor from here and seafloor from the western side, where again the seafloor there is only 20 years old. Comparing the microbial communities that colonize the 23 versus 20 million year old basalt will tell us how the age of the basalt affects the kinds of microbes that we observe on the surfaces after 6 months of being left behind on the seafloor. So that's a general idea of like one of the things that this colonization experiment can tell us.
I see a question asking about the hectocotylus, the modified arm on the male octopus, if that always falls off. And I can say no, that's not the case. There's species where it certainly stays intact through the entirety of the male senescence.
That guy woke up?
Yeah.
Yeah?
It's nice to see it swimming.
Startled a bit, but looks like he's going to make it.
Who's coming first? I mean, like the better views on there is top down here. Maybe you'll see.
Yeah. It's like they know each other, but they help each other.
Got on there, man. Next one. Good.
Nice.
Lock the jaws. No, lock the jaws. Maybe show 'em.
I hate doing that. Then when you need to let go or something. What he's talking about is
So we've got them all?
right on your.
Yeah.
Oh, nice.
That's what that means. If you
Single trip then.
your jaw,
Nice.
Utilizing both arms.
Yeah, yeah.
change it to longer right there, but now you can't accidentally hold it.
And I guess we're transitioning to marker King? Yeah, right around the corner, right? Where the pots were left and the PVC pipes?
Oh,
No, wherever they're shimmering, I'll take it.
this is where we were,
Okay,
where we just came from anyways.
okay.
This is where we're measuring the shimmer.
All right, perfect, yeah.
Just before.
Wherever they're shimmer, that's going to work. I just want to leave these guys as close as possible to a lot of the shimmer and not the background ocean. Interestingly, though, who knows how variable the shimmering is, you know? Maybe next week it's a completely different spot that's just...
That's where you say top four. That could be affected by the tides as well.
Yeah, it does, yeah. It definitely is, yeah.
just a little bit so I can see the end of that ram, and we'll just land. And maybe-
Anyway, for anyone that's just joining us, we are in the middle of transferring a microbial colonization experiment from a place of moderate shimmering to hopefully a place of a lot more shimmering. Again, the shimmering represents cracks in the ocean crust here, where subsea floor fluids, that is, fluids that are generally flowing under the sediment layer that's found here in the East Pacific Rise, emerge to the surface. And those fluids are hypothesized, or actually are known actually, to be completely different from the bottom seawater. That's why they shimmer. They carry a different physical, geochemical, and microbiological signature. And we're trying to understand what that signature is, and how those microbial communities that are venting out in the shimmering fluids differ from the background seawater, and potentially affect the ecology of this wonderful habitat here, the seamount ecosystem. So that's the real goal, or like I guess the rationale behind these deployment experiments. Oh, yes, plenty of fluid too. Yeah, this is nice. Would this be an appropriate landing site for maybe one or two here? Yeah. Oh, yeah. Just leave it. Set them first. Yeah. I understand. Yeah.
Actually, take a look over there. Is there shimmering? 'Cause
Where?
If there is, that might be a decent spot.
Yeah, so one of these is inadvertently landed. Well, we've landed. But
On-
it might be a good spot anyway. So
Yeah, see
I think it looks good to me. Yeah. That one's gonna work. I think that one's done.
Just,
Oh, nice.
I knew when I was repositioning, that's it.
Can we get a frame here? Thank you. Yeah, that'd be cool too. Please, yeah.
So, for prescription?
That would be it's a microbial trap. Deployment one. And then just RHO for Rodocker
the wrong side of this goes on.
Do you want
Sort of looks
slashes?
like, see the hose there?
I'm like, I usually use underscore for everything, but it's the easiest to link. But it doesn't matter. This is not gonna be a CSV file, it's just a
You can't
bunch of
make it.
searchable words, right? Yeah, so we're cool. Just my programming mind.
No, I
How am I gonna
agree. I just
limit
didn't
this?
know what RHO was.
Yeah, got it.
If you wanted to zoom in, we could video it for.
I can control that here?
Yeah. just
Okay.
Yeah.
Well, I guess that way, yeah. Yeah, whenever you started that video grab, that was like the perfect moment. And that would be the 5 seconds to loop over and over in each presentation.
That's what I'm
Yeah,
saying.
heck yeah.
Yeah. You should get that while you're in charge.
Yeah.
Come in under elbow a little bit. This looks the elbow.
So again, just to give the audience a little more background here, the experiment that we just placed on the shimmering water contains a mineral substrate called rhodochrosite, which is actually a manganese carbonate. I hope that this manganese carbonate accumulates over the 6 months of long of the deployment a ton of microbes that like to play with manganese. So I suspect that some of the manganese carbonate will be driven to the solution, and the dissolved manganese will then hopefully be reoxidized by whatever microbes are in the shimmering fluids. And I hope to come back and maybe see some dark spots that would signify manganese oxide driven by manganese dissolution, originally on this pink mineral experiment. Again, the mineral is called rhodochrosite, and that's what we just placed. And we hope to come back in 6 months and pick this up and bring them back up to the ship. Once I retrieve this in 6 months, I plan on extracting DNA and potentially RNA. Both of these polymers are called nucleic acids, and they can tell us a whole lot about types of microbes that were living on the rock, again, after 6 months of deployment, and most importantly for me, the microbial activities that were occurring at the on rhodochrosite versus the other sterile mineral substrates that are part of these colonization experiments.
So pitch out, elbow down.
Good, yeah. We got one really nicely placed one already. Yeah. It's a lot of shimmering fluid. We got Holly got a really nice video, so I was saying that's the one I'm
Well,
gonna.
I got a frame
That
grab.
frame grab.
Just stay
I
there.
got a frame grab on the video, they panned out right when I asked you, so I was gonna say before we leave, if you want to get, but you can find that frame grab and then we should do it while we're here.
Yeah, for sure. Okay.
Or using that.
Yeah. As soon as we place the next one, I'll tell them to slow down for a video grab.
Pause
Yeah.
and
So yeah, so I should be able to get that 5-second video that plays over and over from every PowerPoint. Yeah.
Just communicate inside this way with these.
Plenty of venting right here too, right? Somewhere there.
Okay, we might have to reposition, oh, actually, Chunky did that one.
This is, shoulder up and down.
I think you can control the.
Yeah, I mean, we'll have a little zoom around and see a few potential spots.
Yeah.
Might have to get one of these.
Or like right up here.
You go each that from where we are to this on.
Okay. Do you have a preference on which one was it there?
I'm just going for the shimmering, so
You need one of them, okay.
wherever there's shimmering happening, I'm not going to be against. Yeah. Where was the probe ultimately inserted? Right around here, right? Somewhere on that plot.
Just to the left of the
Yeah.
box.
Yeah, but there was a 10-degree observation here already, right? Okay, yeah, so as long as they're shimmering, I'm happy. Okay. But it's knocked right in between these two guys. That'll be fine. As long as we're not, yeah. It's gonna be cramped quarters there, but.
Yeah, that's where I am now.
It's hard to tell the slope on those rocks, huh? Yeah. Perhaps moving it up a little bit here.
Yeah, just a little bit further like that. Slow.
Right there, yeah.
Which one do you want to grab? What's this one?
Oh, nice. Tim, it's on there.
Remember, keep an eye on both.
Yeah, wherever they're shimmering. I'm trying to look here. It seems that when we zoom in, it's everywhere. Thanks for that, Tim.
Let's
Yeah, so that's the McAllister and Barco papers. Right?
Yeah, yeah.
Yeah, so I can just let them. Yeah.
Try to flip or somewhere else.
Sorry, what's that?
You just, I would just move it to the side or freeze
Go ahead.
it and then let them move. And see
There you go.
for shimmering, see if
Nice
it's
little spot right there, huh?
up there.
Yeah, I think we found a little nook. Yeah, away from the animals. Is there shimmering there? Maybe not as much, huh?
Not much.
There's no, don't know. how close
Down
that
here?
shimmering is.
Yeah.
Hard to tell if that's in front of us or.
Yeah. Wow, these cameras are amazing, but I guess we're still limited to 2D depth perception, right? So I guess that's the next step for an ROV, right? ROV operations.
We've got to try and marry these two up
Yeah.
to take your depth perception.
Right.
I mean, you can zoom in on this one as well to see if you can see.
Yeah. What about this little corner here? Is that out of the reach of the arms or something? Like right over there?
I'd have to maybe move a little bit.
Okay, all right,
Usually do
let's-
that.
Can we zoom in here just to see what the shimmer situation is there? That might be easier. So I am trying to work with the position of the ROV. And again, this might change with the tides, but I'll go to sleep easier knowing that I left there with a lot of shimmer. I understand it means nothing like a month from now.
Yeah.
Yeah. That's cool. Yeah, that looks pretty good. Yeah, I think that's promising. And again, our
Nice little nuke as well.
yeah. Yeah, so anywhere there, I think I'll be happy with. And again, our ultimate marker are these guys. You know?
And it'll
Yeah.
follow you over.
Spot, Gus?
Yeah.
Was it this spot or was it?
One second. Where the heck do we see that? Yeah. All right, one second. I'm dead. Yeah. No. Yeah, one second. My God, this is so easy to lose. Okay. Yeah, I'm not sure if this is vigorous enough. Well, it's there.
It's there, yeah.
Okay. Yeah, let's commit to this place. I'm fine with this. Yeah.
Yeah?
Yeah.
I think it's actually this. The crack just there, right where you are now. I think it's there. Well,
Yeah.
the proper zoom before you lay it down a bit. Sort of hold it in that area. Just lift it up a touch so you can see past it.
Definitely see it coming out of here.
Maybe it was just a little bit to the side.
All right, there it is. I mean, it's, what did we set? Right, yeah, right on top of that rock. Let's use that rock as a marker. I'm not sure what the size of that rock is, but oh, we're right next to it. Okay, yeah, I think that setting it here will be just fine. Yeah. Again, it's impossible for me to tell the slope. Right? Does that seem pretty flat?
I think it's a bit of a slope, to be honest, but
Okay.
we'll see how it.
Guess we don't know till gravity corrects us. Yeah. We're gonna wake up the snoozer too. That's wonderful.
Just keep coming down a little
Yeah.
bit, take the slack off the rope, like that, and then you'll see if it'll stay. Looks good.
1 second, can we do a really close, a really good close-up of this so I can record a maybe 10-second video?
Yeah, I'll just
Yeah.
make sure he's
Oh, okay,
copying
got it.
as
Oh,
long as
okay, I
we
understand. Yeah,
just
yeah.
Catch the robot.
Yeah, yeah.
If you leave your arm up there, that's fine. It's good. Just now, yeah, we'll give him a
All right.
Alright, I'm gonna start
Yeah,
now.
ready when you are.
Okay. So starting now.
Yeah, and this would be a microbial trap repositioning MS mild steel.
With underscore now. No.
Together, yeah. That's it. I mean, whatever. We're doing what, 10 or 15 seconds, and then that should do it.
I don't control the amount of time it's worth. I'm not sure how long it goes, but I think it starts
Do we know how long a video grab goes for on a C-Log?
10 seconds.
pass. I don't know.
No.
I'm sure.
I'm honestly not sure. I
I've
can
done
find
a
out.
few, so there's gonna be something.
Okay, yeah, I guess, yeah, we'll do two.
You just wanted to know how long the video clips are.
Yeah, for each video grab, yeah.
So it's just gonna break the video up into 10-minute chunks.
10 minutes.
Yeah, and so, but like, it doesn't start when you hit the highlight. The highlight just tells us which chunks we need to give.
Got it.
Yeah. So like.
So it's always there. It's
Yeah,
just,
yes.
it's just going back to the larger file.
Exactly.
All right.
Because like we're giving
Just
what,
click it
16
now.
hours.
Oh, okay.
Perfect.
So I've done maybe a few more than I should have, but.
That's fine. Yeah. No, all it does is give you a spot to look back through the footage to find it. That's all.
Perfect, so we've been here about 45 seconds and I'm done, yeah. Yeah. Mhm. Thank you.
Okay, so those two are in place.
Those two are done, yeah.
This couple here.
Yeah.
Do you want me first,
Trying to
shimmer,
work with
so
you guys,
don't
yeah.
have to move first, and then we can move if you've got another
Yeah.
spot.
That looks pretty good, right?
Looks a bit shimmery, I think. I don't know.
Yeah.
Looks like there's a bit of shimmer in front of that rock.
Yeah, yeah.
There's also that spot just to the left of the pot. It shimmers quite a bit. And there's a little hole where there's no occupants. Probably put one of those in there.
Yeah, just up next to that.
Yeah.
Terracotta.
That's where I took that water sample.
That would work too, yeah. Can we try that?
Sure.
I think we can reach it better when we can.
Yeah.
That might be a stretch for. So you do that one, Tyler, you can do that. The one that we just looked at now.
Oh, yeah, it's really happening around the pot, huh? Yeah, I don't know if that might interfere with the pot as the experiment actually.
Okay.
Yeah, so let's yeah, let's I don't know that it'll
There's already one there.
yeah. I mean, I guess this seems like a good spot too, right? Any landing site there, there's plenty of venting. Yeah. That works. Yeah, a lot better. Yeah, I don't think it matters if it's that close to the pot, but we should talk to Janet about that. No? Okay.
I think there's okay, just not.
Yeah,
that
not
right
physically,
above it.
not in physical contact with it. Yeah,
Yeah.
I understand. Yeah.
Not that I-
Beautiful. Seems like there's a lot of shimmering being focused through the PVC pipe. Yeah. How about if we place that one here somewhere? I mean, we can almost just leave it there, I think. Maybe moving it a tad. Yeah. Hard to tell, right?
Is that shimmering before it? I don't know.
It's hard to see on
Yeah.
yellow, but it is shimmering.
There's definitely a lot of shimmer going on around there. Look at this bit here.
Yeah.
You can see on the rope, right next to the white on the sample, so.
Yeah.
Nice. That's pretty good. Were they in shimmering water? Sick.
Yeah. It seems pretty diffuse, but definitely ubiquitous, right? To this entire area.
Yeah, number one.
Yeah. So okay, looking for a standalone spot here for this final one.
The plate's threaded onto the.
Um,
I know what you're talking about. Yeah.
okay, that looks pretty promising for this shrimp part. Yeah. Is there a suitable landing spot there somewhere?
This little hole here might be a nice little.
Yeah.
But can we like suction into the holes just to see?
Sure.
like,
Can we try that?
Yeah, just like a nice like.
Going back down.
Gosh, there seems to be so much back here.
With the big rock with branches on it, maybe? That would be great because then also. Yeah. Yeah, I think that sounds good. And that rock would be kind of nice to see what's living on it.
Yeah, yeah. Got it. It's a smaller little hole than I thought when you said the size of this thing. I thought it was a bit bigger.
More critters living in there than we can see, I'm sure. Yeah.
Are we close the door on the?
Yeah.
Yes. How's that feel?
I'm afraid I can slide.
Going up a little bit?
Yeah.
Try and
Maybe
turn it
repositioning.
90
Yeah.
degrees, maybe.
How many rocks?
Three. So there was one collected in the Rovi lava field. It's very small. And then there's two that we collected by Rovi right now that are like 10 centimeters each. Well, not necessarily. There's one that has a bunch of worm casings on it. And top of about 2 centimeters off before we
Seems like a much better position.
That's good.
Yeah.
the rinds on it.
Yeah, I think that'll stay.
Probably 100%.
Unless, of course, there's a catastrophic rock slide or something. But,
Bigger problems.
right. Okay, I guess I'll have a stick around for maybe another minute or so to get another video grab, and that's gonna do it. Yeah, zoomed in as much as possible just to capture some of the venting. Yeah, I'll give you the green light for the start. Is there a way to zoom in just a little more? Yeah.
Did you take a picture of them?
Yeah.
seeing as much shimmering as the other one, but that's okay. I guess we're taking the same chances that octopus has taken on its progeny. So
But yeah.
If he thinks it's a little warmer.
yeah. Okay, you can start the videograph, please.
I took pictures before chipping
Microbial trap. We're positioning 2. What is this? You can just write basalt. That's it. Maybe we'll wait 20 seconds and then we're free to move on. Next order of business.
Could we take a nice picture in the box?
It's funny how despite 20 HD cameras recording this all the time, I feel like I have to take my phone and take a picture. Yeah, that's irrationality. So that's just my own irrationality. Don't judge me by that. All right. You want to take over? Jorge? Thank you guys so much. I really appreciate it. All right. Three more of these. Yeah, thank you.
Could we check the basket for a minute?
Yes.
Make sure.
You said you're gonna put some out on the site.
Yeah, that's
Stuff like that. And
this last one is the Adamar spot.
we check the basket. Make sure that we just displace. There's one here also.
Yeah.
There's this one here, it's still wrapped in the other century.
Okay, so we want to look at the firebox?
Yeah.
So you're gonna do.
So just make sure that the shoulder's all the way through the back. Yeah. So we index and make sure that that's all the way up. And then the rest of the just fold. And just make sure you don't touch the arm. You know, keep like a little bit of distance. When you elbow in, just make sure there's a small gap between the arm there.
Oh, sorry.
And then from the back,
Or even if just doing,
so you'll have a hard
that's something that's just mid ample time too, it's anything that actually
to be positioned there.
like to be
Shoulder,
the other things that's done.
and then pitch out at elbow in. You put your elbow locked just now, so the index, give yourself a bit of play. That's it.
I've got 3 more experiments.
Remember, trying to match the shape.
I guess an initial communication, just a little bibliography and stuff. And the potential distribution. Had a comparison they talked about.
Yeah, so that's like perfect.
Yeah.
One done.
Little gap. Only thing you might need to do is azimuth outboard, just a touch. Yeah, so you sort of slide more straight forward so we can get the approach out and stuff. So just azimuth round, just a little touch more.
I did that, remember that like chapter that,
Close your jaws as well.
it's like microbial life, ocean process, perfect chapter, so. But I have a lot of that. But I have at least this.
Ended up very nice.
You guys killed it on that. Take a pulse.
Then,
As you know, on page.
Hi.
sorry, hi.
We want to pull the basket out so we can see into the bioboxes and confirm which samples we have and what space we've got left. And then we can also leave it out because there's one more thing to deploy, which is in the rock box on the port side. Yeah, we want to look at the porch. it does look like that's open and that's open.
Yeah, there's that mark.
Yeah, there's a marker in there. Why is there a marker in there? What's it a marker for?
I don't know, it was there when we started.
Oh, you know what it is? It's from Gus's deployments. Okay.
Oh, so we should.
We can get rid of it later. No, it doesn't. No, it doesn't. We don't need to deploy it.
When it's when you open it comes out, it took us about half an hour to put it back in.
Okay.
So once we open that,
Yeah.
it will float. And
Okay. We can just move it to over in the other bio box or the other rock box if we need to get it out of there.
okay,
So that means that, but there's broken samplers things in here.
is
The cap.
the cap from
Okay.
the push course.
Yep. Yep.
But we can put some other
Yeah.
things
So we
in
can
there.
put something here, here, and here,
Yeah.
and then here, and these. Yep.
And also like a rock with polychaetes or something on top of the nets.
Okay.
We can have a rock that's not gonna go anywhere.
Yeah.
We got plenty of space.
Yep.
You see the octopus
Yep.
is very, so I don't think it will escape if we open it.
Okay. So what the thing we need to deploy is in the porch, or port side aft. It's a white bag, right? The wood exposure experiment. It's white.
But
Yeah, it's
I
under.
don't s- I don't see them, that's what,
Did it not make it in here?
I don't see them.
Because it was left on the. It was left out last night to be put in there. Do you? Can I look over here? I don't think I can see in there. Do you have the ability to zoom in on the brow cam to see what's in that box with the yellow net?
Probably, this is only like a.
Yeah,
tilt,
okay.
So it'll just
Doesn't-
zoom straight, so we can't get the.
Oh, it doesn't look like it's in there.
Underneath the yellow, there was another like light.
There was? Okay, then that's what we want. So we can go ahead and whichever manipulator you want, like take that yellow net out so we can get the white bag from underneath it, and then we're gonna deploy it in that shimmering water.
Okay.
Which we can also reposition if we need to.
Try to use easier stuff. Might try and reposition. What do you think? You want to try and reposition a bit?
So my initial thought is like I'm gonna nudge the yellow up and see if I can grab it. If that doesn't work well, then for sure I'll just grab that net and pull it out of the way. I think I can get it.
I don't think we're going to get any more porch out at this
That's fine.
place.
I think I can do that.
I'll try.
It's in there. Sorry? I said it's in there. Oh, okay.
It's in there.
Yeah. Jill said he saw it earlier.
There's this now. Oh, this is the sound. It's down, my God, it's crazy.
Lots of blood.
The little monkey fish just came right up. Oops, this is what face. Tyler, was that your first time on the minute?
Pretty much, yeah.
Nice.
A little bit earlier too.
Okay,
Thanks.
nice. You did well. Yep,
Close to a marker to make sure we find them.
right here. So inside this white mesh bag are some wooden blocks. And the purpose of this is to study animal colonization of these wood materials representing or replicating kind of wood falls, natural wood falls that happen in the marine environment, which are often colonized by animals and could potentially be stepping stones for colonization. And so by putting out our own wood pieces in a targeted way, we can study how those organisms colonize the wood under specific periods of time and compare that to what colonizes other substrates that we have down here.
We just want them close to the other things so we can be able to pick up everything it's assembled.
But we do you want them inventing?
Yeah.
Yeah, right. So then we want to put them very specifically over in this area, underneath the pot. Holly, can you take a note of this deployment, please?
You want a frame grab or just a note?
Both. So frame grabs are always good, but then using the. You don't need to worry about that if you just put save for frame grabs.
Oh, okay.
Doesn't require a description. And then under the deployments button,
Oh, ID.
it doesn't have a specific ID because it doesn't have a number on it. So Beatrice, yeah, in the future we should label these in some way so we know what they are. Yeah. So just wood exposure experiment number 1. Yeah. Great. Rock box. We're at marker K, and it's inventing.
In like 2 words.
Yeah, inventing. Yeah, inventing fluid, however you want to describe it. Perfect. And can we zoom in on that to see if the shimmering water is coming through the bag? Great. Can you get another frame grab of that, please? Yeah, great. Thank you so much. That's great. So now we are done here. And we can go ahead and pull the box in, the basket, porch. You guys call it porch or basket?
Porch, yeah.
Okay. And our next activity is we're going to go back towards the marker E area, towards the top of this outcrop, where there was those big coral fans and sponges, and we're going to try to get a few more samples of animals. Olga, will you guide which ones you want? Okay.
looking from here, we can take advantage of like this.
Oh, well, hold on.
sea
Can we hold on? Don't fly away yet. 1 second.
pens. These sea pens that we collected yesterday, but
Yeah.
they were destroyed.
Oh, they were destroyed. So
Maybe
you want
we
to get
can
another
take
one.
at least 2 more or 3 more in order to see if they hold.
Okay. Do you want to do it again, picking them up and putting them in the coral quiver? Okay.
Like 2 maybe.
Okay. Apologies. These sea pens, we're going to try to collect a few of them to try to increase our chances that one of them remains intact. They're very delicate. So picking them up with a manipulator towards the base and then trying to carefully bring them into the coral quivers. So we'll try to collect 2 or 3.
They get those easily. The problem is that they get this one when they go up.
Is it okay if we put a couple in the same quiver? Okay.
But just that being a bridge, I'd rather be away from the bridge.
May I? Can I do some zooming on while you're talking?
yeah.
Thank you.
I'm quite sure it's safe, but I just thought this was an opportunity.
So we have
To Ray, and just kind of keep that in account when we invest, we don't want to invest uphill side.
Is this a target species that we would want?
Okay, gracias. Yeah, it is. Spider. So nice of
Also, if that is a ridge, just imagine if we went up to it and went over the
Probably the slurp would be the best way to get that. Yeah? Okay. Before. Oh, one moment. This mobile species here, this sea spider with several commensals, we might like to try to get that with the slurp also. The slurp section. So I'm not sure what would be your preference, to try to get that first or to try to do the sea pens first.
Since he's running away, I say let's get him.
Okay, let's try to. I'm gonna. Yeah, I'll let you do that.
Yeah, that's good. One portion and then, I gotta do it. Which tube are we in on that can? It looks like we're between, actually. Maybe index it and line it up. Try not to go full speed, go like 3/4 speed.
Can we aim it down when we do that so we don't blow it all over the animals?
It'll only sucks, but you can do that.
Ah, I see. Okay.
Right now we're having issues.
Yep, no worries.
Try it in 2 different directions.
Yeah.
So you're getting an inside shot of the ROV here, turning the camera around to see the suction sampler.
Okay, I'm gonna have to give it a nudge. Just keep an eye on, is that camera tucked all the way back?
This is like watching someone try to scratch their back.
Look left and follow the end here.
Pushing the camera there, mate.
Okay.
Trying to go ahead and index.
Ooh, all right. That
One more, line it up with one, but don't go full speed. That's what causes that to happen.
It's not moving at all.
Yeah, it's, we can't go full speed. Try it again. Try it again. Stop moving yet.
No.
I think it's see it spin.
Try the other way.
Okay. There it goes. Try the other way again. Okay, yeah, stop. Again.
Try the other direction.
Yeah. There goes. Give it a little bit more.
Same direction.
Yeah.
No.
There it goes. Got it. Alright, what are we lined up with now?
We're on
That
eight.
is- That is 8. We're
Yeah.
in 8.
okay.
Go ahead and. Oh, you're flowing.
Yeah.
It's going in 8. Perfect. Where's our speaker?
You'll be away.
There it is.
So before we scoop them up, we want to get some close-up highlight video of that animal. Is it?
You might never have a scratch now.
Oh, yeah. 100%.
You want to get the video now, and then we can
Sure.
reposition and you can.
If you can just, yeah, log that we're recording. Or even just a highlight is fine. Okay, that's good enough. I don't want to let him get too far away. Yeah, and then whatever you need to do to get in position to collect that.
Yeah.
Yep. With this wide field of view, we can see lots of different animals here, in addition to that sea spider with his friends that he's carrying, or she is carrying. Also got a little crustacean next to some of these octopus. Many, many different sea pens in the field of view. What sample number will this be, Holly?
It's 051.
Okay.
This is going into slurp chamber 8.
Did we change 8 or is it still 8?
It's still 8.
Okay. I'm assuming all the sediment blew out of there.
Looks like he's defeated us. Drop the biobox.
Yeah, we could drop him in BioBox 2.
Okay, 2, that float will float up as well, and then we'll have some trouble getting in there, but just letting you know. You can still do it, but
Yeah, we can put it in and we can take that float out if we need to.
okay. That's more trouble. He's about to go in there.
Wait, there you go. We can move forward.
Yeah. You're just getting a better grip.
Can you try to shake it at all? Would that help?
He's down there.
Okay. Okay, well then,
He's turning the flow off, and I'll
yeah.
try to shake it in there.
We'll try to deposit our collection on the B side.
Just open it back so that I can.
Okay, stop the flow.
Can we open the lid more so that he can get it more vertical?
Yeah, I'm just trying to keep that full.
Yeah.
We're gonna have to. I have variable movement.
Yeah, the float is not essential. We can just get it out of there.
I'd actually click on the float. There you go.
Oh, this one doesn't have the divider.
Start closing.
Those arms are sticking out. Wait.
Okay, close the other bio box. Close the other bio box. I don't want the lid on that bio box open.
It is closed. It's got the rope is,
Ah,
that's what the octopus is attached to.
I see. Okay. It's okay.
But don't worry.
Yeah.
The escape is alright, it's okay.
Yeah.
It's fake aqua.
Yeah. Okay, so we can go ahead and stow the slurp sampler and bring, we'll reposition to get some of those C-pins. So what are the other, besides the CPAN, what are the other things that you want?
I saw 3 different corals,
Okay.
but I understand that we are moving somewhere else. We are not going back to the places we mark, no?
The places we marked this morning,
So that's the,
is that what you're referring to?
yes, but if you want to explore somewhere else, we can go somewhere else.
Oh, no,
As we can see
The priority was to finish the things on the dive plan. And then once we're done with that, then we would go try to do what we didn't do yesterday, which was to go to marker A. But that's just to fill the time until we dive. So getting these samples is a priority.
So I think that they marked some places, but I'm not sure.
They were marked yesterday when we were here?
Not today. Today when we were doing the transect,
Okay,
it was some places.
I can find
where-
it for you.
Uh-huh.
It's also should be logged up there. And we're back. So we don't, what we're going after are these skinny pink things here. So wherever is a good spot to line up to try to get a few of those.
Some lower down so that we're not above the pots and stuff.
Okay. Yeah, there's several there. That looks great. Why?
It's a group,
Mm-hmm.
some together.
Yeah, that might be easy to just grab several.
Just grab.
So our interest here is collecting some of these peach-colored sea pens, so we can try to identify what species they are. Got a Trevor, are you called a pilot in training?
Tyler, yeah.
Tyler, sorry, not Trevor. All right, we've got a delighted. We have some additional training going on this expedition, in addition to our scientific team, also our operational team, getting experience doing this deep sea research. You get a frame grab of this, please, Holly, before we collect it. Yeah, frame grab is just like to get it.
You want frame grab of this first? before
She
we
just
grab
got it.
You got it?
Yep. See some interesting little baby, what look like demo sponges in the background. Those little gray things. I think that's what those are. Yeah, okay.
I intend to use it there because I found that button.
These specimens are being collected for the Museum of Zoology at the University of Costa Rica and will be characterized by some of the experts that work there. Yeah, and we'll actually try to collect a couple and put them in number 4, so you don't need to put the lid on right away. And you can see once we collected the sea pen, the polyps closed up. It's coral quiver number 4. And then, did you get this recorded as a biology sample? Okay.
Try and like sort of lift your arm up so that the jaws are pointing down the way, and then just rake
Should
it
just be
across
coral.
Have it like that. Rake it across the side. Then you'll have the clearest edge. You won't have your arm a bit clear of the other stuff. You know what I mean?
I see where it's hanging on.
Oh yeah,
There's a little string on the top.
yeah. Watch your top camera as well.
Some really sticky threads there.
Another one of you as well.
Yes, please.
How many? 2, 3?
3 is good. When we line up to get the next one, if we could get a really tight zoom shot so we can see the polyps out.
Do you think of it? The one right behind you there.
Yeah, we could also, this bunch there,
I'll zoom in on this one.
yeah.
Just take the T4 out of the picture a little, Stephanie. So that
We'll grab a frame grab of the close-up. Is that as close as in as we can get?
fine, make the light a bit.
Okay, yeah, great.
That's as much zoom as we can get, yeah.
Okay,
Is that lighting what you want?
no, he's still working on it. It doesn't hurt to just take multiple.
Yeah, I have that.
Okay.
Good.
Yep, that's good. Okay, you can go ahead and try to collect. And you can pull out as you need to. Is that a sea dandelion? Do you know those?
No.
I don't know what they're called in Spanish.
What is that in English?
Sea dandelion? Yeah, I think that's what it is. It's a siphonophore. Pretty sure that's what it is. We'll zoom in on it after they're done. Mm-hmm. I'm not sure if it's what it is. So this will be a little tricky because we're not going to be able to distinguish the different samples in the bucket because they're all the same.
If they put it in, head side down,
It's kind of get jumbled up. Yeah, so I think we just call all these the same number. There's just multiple individuals that reflect it. Otherwise, it's just going to get complicated. Yeah.
Come straight up and down, yeah. Hook. Close your jaws a little bit again. It's just a little bit.
Yeah, this is some expert manipulation by a pilot in training. Yeah. And the sticky bit. There we go. All right. One more, please. Actually, before we do that, do you mind if I take the camera for a moment? I want to zoom in on this other animal that I think we see here that I don't think we've seen yet. So I believe that this, oops, you're very sensitive here, Mr. Camera. I believe this is a sea dandelion. It's a type of siphonophore. I'm not exactly sure that's what it is, but that's what it looks like to me. There we go. Okay. Yeah, what would you like to pick up this time, Tyler? We've got several right here in the lasers. I don't know if that go in. It's okay if we get more than one. If that looks like a good spot to you, or if you want to get something closer to the minute, that's also fine. Yeah, if you. Part of the reason we're trying to collect a few different specimens is because they're fragile and we're increasing our chances of getting something good by getting a few different ones. So yeah, if you think you can get around one or two, that's great. Yeah, maybe just the two, but whatever works for you. Yeah. Great.
The two.
Nope, looks like you did get three. It looks like you was able to collect three, so that's great.
Perfect, that's great. More chances to get good shape.
Yeah?
Maybe that would be the heavier end of the potential solution for one more drop in potential. See if it's going to be long enough, or else I'll get into the other two things. It's going to take short there. I'll have to send a jack on these two things, and all we'll do is we'll pipe them all together, so we'll listen to it.
We're doing the games too. Yeah. Yeah. Less possible.
Sorry. Okay, so one of you should get dinner, and I'll approach over to you go along, or if one of you wants to go, you can keep going.
Where do you want to stay?
All three of us need to get dinner in the next hour.
Do you want to? Oh, no, you can't.
Either way. No,
Yeah.
I can.
I'm fine.
Okay.
An extension.
You can go for dinner.
Yep.
I can take
Yeah.
you.
Okay.
Yeah, so just look behind it, there's a yellow mark.
So Una, sit here on the screen. They put these green dots where things were observed. It's 1, 2, 3, 4, and that was it. At some point they were dropping 20 points of
Yeah, we're just zoomed in, if you zoom out, there's, like, what are you looking for?
they were putting dots where octocoral were observed, and she wanted to pick out examples.
Oh, okay.
One of them,
Yeah, so the octopus marker, what we can do is we can look at, do you know how many markers ago that it was? We can look back for a
I
while.
don't, and I couldn't look and see why we
No,
were
those
go
guys
back
just,
like 10
you
minutes.
see it in there, sitting on the.
Yeah, it's bumping.
No, so you can, because they log the events where there's all these things were, so we have the over special works. Yeah, so maybe we can start with yellow spiders since it's on them. Yeah. So we'll fly there next. I'm thinking we'll be back into another room. Can I have my water bottle?
Where is it? You see something?
Yeah, I agreed to picture your store from this camera down here. So I'll follow you in just now, but get used to it.
Okay, so good evening, everybody. I'm Dr. Odalis Cabridi from University of Costa Rica. I'm here with Gwendolyn Matamoros from the University of Costa Rica and Holly. And Holly from Schmidt Research Institute. And we are going to fly to collect some sponges and octocorals. We are going to a site that we already marked with waypoints, so we are moving there. Buenas tardes todos, yo soy Odalisca de la Universidad de Costa Rica y me acompañan Wendy y Holly, que van a estar tomando los datos de las muestras que vamos a tomar de octocorales y esponjas. Por supuesto, contamos con 2 pilotos muy experimentados, una persona que se encarga de multimedia y otro técnico que se encarga de las comunicaciones también. We count with excellent pilots, very experienced, that are able to collect very difficult samples like those that we are going to collect now. We also have some technicians that are in charge of the communication. So you just enjoy our trip. We have to go to the first point where there is a yellow sponge. He has the coordinate, the coordinate of the first point. Ask him. At the place where we have to go for the yellow sponge.
Yeah.
Okay, you can guide them to go there?
Yeah, I think it's just right underneath the vessel. Is where the sponge,
Okay. So that's back down, so we have to go back down?
Yeah,
Okay.
yeah. Yeah. Don't ask me for directions because I get lost in my own room.
ROV, Pete, ROV, go ahead.
Hey, Jason, I'm just going to move your waypoint to the south view. Where's down there?
Okay, yeah. What speed would you like?
What are you doing right now? 0.2, maybe a little faster if that's all right?
Yeah, got it.
Thank you.
Just reverse it quick, huh?
You happy to drop down to go to yellow sponge, or would you like to look for another yellow sponge on the transit?
No, just go to that one that we already spot and collect it and take a good picture first and then you collect. This is what we need. Maybe later we will have something more interesting than that.
I'll come up and I'll just reverse back. What do you think?
Yeah, usually if something feels like this, I'll just do the reverse and slide down. So you still get a good image.
Yeah.
Or you can go sideways and then. However you do, that's all I can.
So as we are moving, you can observe some octocorals that we later be collecting and some sponges. Then we will collect some of those. Conforme nos movemos a través del fondo, cerca del fondo, vemos varias colonias de octocorales y esponjas que vamos a recoger después, ya que son uno de los grupos en que estamos interesados. I think there, yeah, it is. There it is, yeah.
That's not the one, but we got a different
This one, we can collect a piece of that. First, we can take a very nice video, close up.
If that's okay, I'll stop the ship move. We're not gonna go all the way back down to the same
Okay.
time. If we just collect this one, you okay with that?
Yeah, but we have to.
Hey, Jason, out here. We actually found,
You can do it here.
so I'm just stopping where you're at. That's right.
Hi, Adam. It's Pete. Jason just gone for dinner, but no worries. Yeah, I'll come to a stop now.
Thanks, Pete.
Cheers.
Wow. Come on. Hay un pinoide. Ah, sí, no, ese yo creo que es un isis, pero ahora lo agarramos, sí tiene un pinoide. Sí, octocoral nada más, ya. Sponges are very interesting animals. They are very important in the deep bottom. There are some of the organisms with the octocorals and with the black corals that made the 3D structure where other animals can live there. In the case of sponges, they are made of single cells. It's like a multi-cell organism, but each cell has functions by itself. And then it works like a whole big organism. Las esponjas y los octocorales y los corales negros también son los principales organismos que conforman la tercera estructura o la tercera dimensión de los fondos oceánicos profundos.
Scallops getting caught now. I'll maybe do what you said actually, just come down with the torch first.
Yeah. Sure. I'll give you a beginning.
Can we hold
Oh, yeah.
this for a minute?
Oh, that's right. Yeah, you can-
Oh, sorry.
Yeah, no worries.
Will we be holding still on this for?
Yeah, we're going to. We're just repositioning.
Okay. Yeah, I just wanted to grab a nice photo.
Okay. We can collect the others in the same spot maybe, or later. Yes, to identify the sponges, we can do it just with good pictures that are taken for the team. And then with small piece of the sponge, we can study the spicules that make the skeleton, and they can give us an idea of the species. Also, we can do molecular studies and compare with other species that already exist and know if we are dealing with a new species. So just with a piece of the sponge, we will be able, and these fine images, we will be able to do the taxonomy.
So, left and right and zoom in and out.
Yeah, oops. It's very, okay. Like here, maybe? Okay. Okay, I'm ready. Yes,
Do you
right.
want just a snip of it or the whole thing?
No, snip, just a piece from like here, maybe if you can. Oh, it could be one of these branches. Yes, it would be better if you can. I was talking about the upper side, but I know that you can. Thank you.
Yeah, Rach, is it okay if I bring the heading 20 degrees to port?
Yes, sounds good.
Awesome, thank you.
Where's this going? That's a good question. Let's do that first. Because I thought that one was open, but what's the name of the Octopus? That's open. He's got that. Let's go to
Yeah, I think it's 6. 6 okay?
Yeah, yeah.
It's okay.
Are you talking about the quiver?
The quiver?
Six?
Yeah.
We haven't used 5 yet,
Okay, let's put 5 in.
but either's fine. I think we should wait. I don't know
Look at that road. It almost looks skinny. Closer to the base.
what it is here.
Yeah, I was gonna say, it looks like, did you go in on this? How did I come straight down? Yeah.
But we need to, oh yeah.
Now the pilots are deciding what is the best way to collect a small piece of the colony of the sponge. Los pilotos están decidiendo cuál es la mejor forma de recolectar una parte de la esponja para los estudios. Good.
Somebody needed a shower exposure.
Number 5. Number 5, yellow sponge.
You wanna go get some dinner?
You finish?
Yeah.
So quickly? I just started. So Dr. Beth is going to take you from now on in the collection of the octographs.
Okay. Did we find that?
All of them are closed in this same area.
Oh.
So you can take some pieces from there. There are at least 3 species around there.
3 octopods.
3 different little ones.
just, I put it here. Okay, do you wanna get, you wanna do this and then you'll get dinner?
Just
Okay.
a little, just, I will go later.
Okay.
It's fine. Yeah, just to show you.
Yeah, yeah.
More.
Okay. You've worked with these before, you know how spiky they are?
How to,
I said you've worked with these before, you know how spiky they are?
you have to use the gloves.
Yeah.
Because they are very, silice
Mhm.
spicules.
Mhm.
They get into your skin, so you have to manage them very carefully.
Mhm.
And lift them.
And you have those gloves?
Yeah, you have.
Okay.
We have
Okay, good.
normal gloves.
Yeah,
yes.
okay.
Yeah. And we use the gloves instead of the hand.
Mm-hmm.
So it's-
But maybe I will, yeah. Okay. I'll go fast. Well, waiting until they put the lid on before I write this down.
You wanna get some dinner?
Yeah, I guess I'll go.
Okay.
We noticed all these are, the same location. I don't know if that's accurate. I hope so.
Yeah, wait for you to start this way.
Location is not in the, I thought location is supposed to be where it is in the.
Nope, it's, where it's. That's fine, don't worry about it.
This, where it was collected, not the bio. So these are all, are we still at waypoint? Still waypoint, are we not? Are these all? Where this one?
It's a window into the sponge's soul. All right. So I think we're just going to fly back so we can see some of those other octocorals in the area. And
I didn't expect that.
yeah, and it's got some hydrozoa on the side.
Okay, you said back, I wasn't here, so back this way?
What was the question?
I wasn't here earlier, so by back, you mean back down this?
So sorry, no, just like pick off. See how there's like an octocoral right in front of us?
Yeah.
So I just want to come off this ledge so that Ora can point out to me which sample she
For
wants.
example, this one.
This,
The
yeah?
one branch.
Mhm.
And we can take first the close up.
Yeah.
And then a piece from the top.
And is it just this one, or is that, for instance, the same one?
It's the same one.
Yeah, I think
But-
that will be easier to sample.
Yes, I think so. Maybe
And
it's
there's
not
that yellow?
big. If it is not big, you can take the whole thing.
Yeah, there's a small one over there.
That one, maybe.
Okay. Do you think
No,
these are?
then maybe we can collect the whole branch. Because there are many around.
That's going to be hard to put.
To put it on.
Yeah. So I think snipping.
Like 20 centimeters long, maybe.
Can we have the lasers on, please? Oh, there they are. That's 10 centimeters. So this is probably a meter long.
It's long.
Okay, so this is one of the target species. What's the other one?
This is the other one is the big one.
The big one.
You could have a piece, just
Okay.
some branches.
Okay, so you want a piece of
Some
this one?
branches of this one.
Okay.
Yes.
Okay.
And there were another one, but I don't
Probably by the time you get back from dinner, we'll only have done those two.
Yeah, but
Yeah.
when it's going for dinner now, I can stay a little bit more, maybe.
Okay, so I think let's try first with this one since it seems nice and flat in front of it.
Fíjate, that is different, the small one, that is with thin branches.
This here.
Yes, because it's very abundant and we would need the ID for the analysis of,
Okay.
of the transits.
Yeah.
Yeah,
Okay.
This is why I'm thinking also.
Okay. So 1, 2, and 3.
And if there are three, this is what.
You said the first one, sorry?
So the 3 things we want to try to get, either these long sticky ones. So it could be that one, it could be that one, it could be this one.
Okay.
We're going to try to get a piece of this, and we're going to try to take a piece of this.
Okay.
So however you think you want to set up. Yeah. Hopefully we don't have to sit on the sponge.
Yeah, I'm trying to
Yeah.
sneak in here.
And we have to be mindful of the heat flow probe here.
Yeah, I was watching that earlier. It is a tight fit. That's quite a reach over there. You gonna be able to do that, Adam? That sponge is to my left. I'm just trying not to just sit on it.
Answer to this guy.
Yeah, this big one here, also
That's why we're gonna
right
just
behind it, that little spider
use
right there.
something.
Oh, this one here, sorry.
Yeah, so there's 3 things we're gonna sample. This big one that's off to our left. Yeah. That one that's behind it a little bit. Okay. And one of these skinny one, there's 3 of them, but we only need 1. Okay.
Yeah, and we're not going to collect the whole thing. We're just going to try to collect, like, snip the branch.
I'm trying to get you in on this guy right here real quick since we're here.
Yep.
Yeah, because your porch is up, so if you get it nice and close to it.
So we're trying to do some targeted sampling of pieces of these coral colonies.
Yeah.
Which one do you want to try for first?
We're gonna get
You want to try to take the bigger piece?
Yes.
So before we do that, we'll just want to set up to get some good stills.
Okay.
So there's a question asking who is narrating. So right now it's myself, Beth Orcutt, from the Bigelow Laboratory for Ocean Sciences. Before me, sitting in the hot seat for a few moments, was my colleague Oralisca Bridi from the University of Costa Rica. She's an octocoral expert. That's part of the reason that we're collecting these samples, so that we can try to identify the different octocoral species. And you can see here a very large octocoral. This is probably at least 2 meters in width, probably extremely old. So how do we want to do this? We can
fix this.
One moment. So we've got that coral quiver, which isn't that deep, but we would like to get some branching structure. I'm wondering if trying to snip in here might be suitable, because that's what, it's about 15 centimeters.
The third big branch there.
Somewhere in here would be great, because that gives us a couple different branching pieces. So before we do that, if we could zoom in on some of those polyps, get really close shots while they're open. You want to take some frame grabs. So push the frame grab button, and you can just press the frame grab. No, not that one. It says highlight frame grab, yeah. And then just push through. You don't need to type anything. The point here is to try to collect some photos quickly. So you can do it multiple times. And then when we are taking it, then you press that one. Yeah. So you can see here how the octocoral get their names. Can you see the sclerites there? Yeah, so those little white things towards the base of each polyp are called sclerites. They help with the identification, as I recently learned from Oda. And these are obviously not obviously, they are filter feeding organisms. So their polyps are out to try to collect particles. Okay, we can come wide on the camera. Yeah.
Ready for grab?
Yeah, you can back out and we can try to collect this. I'm hoping that if we just take this smaller piece, that it will fit in the coral quiver. Maybe my eyes are bigger than your manipulator. In the background, you can see a yellow crinoid attached to this coral. We're getting some commentary from some experts on Pacific Ocean corals, suggesting that this might be part of the J3 clade. And you can see that the polyps have now retracted as we've been interacting with this coral.
Nothing's moving, so you're not, no, you're not touching, you're fine.
Just at the end of my reaches.
So if you could again take some more frame grabs as we're getting this set up. Okay.
Yeah, I just.
You can see the base of this coral and how large it is. Yes. Just the number.
Sometimes when you freeze it drops. Yes, I know. That rock did that to me. Oh, sorry.
So we're getting a suggestion or confirmation from a colleague, Les Watling, that we believe this is the J3 clade. Thank you to everybody that's joining in with us. We have a few more hours on bottom for this dive here at Dorado Outcrop. We're getting some of our final animal collection samples for this dive. We're at a part of the outcrop that is facing more to the south and with much steeper relief. And this area seems to have more larger sponges and corals than are observed in some other spots of the seamount, giving us an indication of the current and the food delivery in this area. Yeah. Just the right size. Nice. Well done.
Do you want the rest of them in there too, or can we close this down?
We can close this. Okay, J3 Octo Coral. CQ6. Yeah. Is that one you want to try to get to? Okay. We'll try to get that next since it's right here. Go ahead and open up number 7.
And so the next one's the one behind?
Well, let's see. So,
It's this guy right here you wanted to, right?
yeah, so we talked about that one. Come back out. We want one of these. This looks like almost the same thing. If we pan to the right,
Yeah, the 2 is to the right. There's 1 and 2.
Yeah, there's 1 and 2.
The third one was to the left of this thing.
Yeah.
But if that's the same.
let's zoom in on that one, the small one, so I can see what it looks like. Okay, so another type of bamboo, it looks like. Okay, and then if we could come wide and then try to get a similar tight shot. I know it won't be exactly the same, especially with the manipulator in the way.
Sorry.
It's okay.
Is that my hint?
No. Yeah, do we think that's the same? It's okay.
That's full zoom. I'm trying to focus it. There we go.
Do you think that's the same thing as what we were just looking at? It's hard to tell with the polyps pulled in, but they look stickier.
Yes, I think it is different from the other one.
Okay.
That's no problem. We can reposition over
Yeah,
here
so
and
that,
get this.
the other thing that Oda was talking about that we haven't talked to you about yet is if we come wide, there's, for example, a yellow one here. There's also some right below us on the starboard side. You can see them in the brow cam. That's another one we're thinking about. So I'm telling you that now as you're thinking about repositioning so that you know what we might be trying to get.
That one's gonna be quite reach for you, isn't it, Adam?
Yeah, I don't think it'll reach that. Well, maybe. You try.
Yeah, that one up there.
Yeah, it's too far.
Okay.
The porch is out, so it is. Yeah.
Yeah.
Well, let's try. Let's try.
And you were like stretched out. Getting to that.
I don't think it'll hit it here, right?
Yeah. Yeah, so if we maybe back up so that we can get that, we can also think about some of those yellow ones in the front too.
Alright, well, we're here. Let's just move forward and get that guy.
Okay? Yeah, either one. It could be that one or this one.
But right now, we are like here and this, they are here, not here. We are watching down, not the
this one we're watching down, this one is in front.
I know it's a little to the left there, but it's good.
Did you want to enter first?
Yes, please. So can we take some frame grabs, please, Wendy? Okay, and if we could get a tight shot, please.
What do you? There.
Okay. Do we get a frame grab of a closer shot? All right, we can come wide. And yeah, so probably just I don't know, snip somewhere in there. How deep are those coral quivers?
What is it,
25?
a foot? What's that? 4 quivers? Yeah, hold that.
Okay, yeah, so
Maybe a little less.
I'll stop. And yeah, I still got you too far out there.
It's heading.
Yeah, you're in the
Yeah.
4 quivers.
What about if you just take off all the head and just rotate just ever so slightly with your cruise on? Just rotate a little bit.
You want me to go to the
Yeah, I'll watch
right?
Yeah, or, yeah, rotate to,
Heading left?
yeah.
So part of our interest in sampling this particular specimen is that we believe we collected something similar yesterday, and by having more than one specimen, we can see if there's actually variability or similarity in the different groups that we see that have common morphology. And again, all the samples that are collected as part of this expedition, all the animal specimens will be archived at the Museum of Zoology at the University of Costa Rica, where we have some of the world's experts in octocorals who will do some of these descriptions. Very nice. Okay, order, you have one more bucket left. Which one do you want? The long one or the yellow one? We can do it also with the slurp.
Can we see the yellow one?
Yeah, we can zoom in on it.
To see it?
Yeah?
Maybe the yellow one could be with the slurp.
Yeah. Now we could cut it and then slurp it. Yeah. So. What sample number was that? 54? Okay. So before we do our next collection, we want to spend a little bit of time looking at the yellow corals to try to figure out if we want to put, which one we want to put in the coral quiver and which one we want to try to slurp. Yeah, so these right here would be great to look at.
Yeah, this is an hide.
Yeah, that's what I was wondering if that's what it was.
I don't know, just gonna put the other part.
Okay, so get the long one. Okay. Do you want this at all? Okay. Okay, so we've confirmed that it's a different species, not one of our target species. So we're again going to come back to this long whip. Oh, you can see the sea star. Thanks for that tight zoom. If we can get some frame grabs of the polyps here. Okay, we can come wide. And same thing, let's try to just get like a 20 centimeter sample from that top.
Did you get a full screen grab?
Yeah, can you take another frame grab? Got it. A little bit lower. Yeah, that's probably good there. Thank you. Yes? Yes. Yes,
After this, you have to guys somewhere else.
we don't have to, if there's something else you'd like to look at.
With this side.
Okay.
But if you see something else in the back,
Mm-hmm. Yeah, we're gonna go try to look at some things we haven't seen before. Might have to push that down a little bit with the lid. There's ice cream for dinner tonight, Oda. There's ice cream for dinner tonight.
you're in charge, but
Okay.
it's ice cream.
Yeah, are you gonna have dinner? Yeah? Okay. You wanna come back in the logging seat?
These, sometimes these floaty ones,
Mhm.
this, will fight us a little bit. If he can get it positioned in the center again like it was, sometimes the best path is to get the lid
Yeah.
and push it.
Yep.
As long as we're happy we're not smashing it or anything.
Yep. I agree.
Okay. Actually, I think we just got it. So. actually be bottomed out.
Yeah.
Is it the end of the world if we put the cap on it and the last 3 inches kind of poke out?
It is not the end of the world.
Mike is determined. He wants it. There you go, at least it's tipping back in.
Get all that? Good.
Yeah. Yeah, I don't think you got any of it outside.
If Adam was doing it, he would just pull the bungee and let it go.
All right, so I think we're done with operations right here. We still have a few slurp samplers that we can use if we find samples of opportunity. But we have completed what we planned for today, which is great because we didn't finish what we were meant to do yesterday. So can we zoom out on the nav map? And I'd like to talk about what I'd like to do next. Even more. So one of the plans we had yesterday was to go to marker A, which is even further off the map still, up to the northwest. I don't know if it's still marked or not. Well, even if it's not on there, my goal is to go explore to the northwest. Here, so what I'd like to do is swing around this side and go this way. And we're just gonna explore, we can just follow a contour up to the northwest until the dive is over.
Yeah, the top side of the contour of the bottom.
Yeah, exactly. So the depth we're at right now is probably fine. And we'll just follow that contour around this way. And then we'll just keep going to the northwest, kind of following the trend of this outcrop.
Okay, sounds good.
Because we haven't yet explored that part.
I was about to ask if that
No, we haven't. We have done it before in the past, but not on this dive yet. Not on this expedition.
that last sample
Once I get to marker E,
was,
I know I'm kind of at the peak of this, and we'll start moving the
Yes.
vessel.
Yes.
off the window.
Yep. So for our audience at home, we're currently exploring the small Dorado Outcrop. It's a small outcrop of rock that's about 1 kilometer long by 0.5 kilometer wide, trending northwest to southeast. We're currently on the south side of the outcrop. And most of the exploration we've done so far on this 2-dive series is explore the southwest face. And now we're going to go around to explore the east side and head up to the north. In our prior expeditions here, the southern end was where most of the venting was observed and the highest density of animals. We just want to see if what things look like as we head north, how the biodiversity and the abundance changes. Thanks for that, Alex.
Yeah, I know it's fun showing off the maps.
So as we're going along, maybe you can see these very delicate, skinny, pink, peach-colored animals. Those are sea pens. There's a really high density of sea pens here. We also have these white Venus vase sponges going by in the imagery. You can also still see some octopus, preferentially locating themselves in areas where there's not as much sediment on the rocks.
Asking if there was any plan to move current deployments to spot with warmer fluids.
I thought we already did that. It's marked off on the thing. We did it.
Okay, good.
And if we didn't do it, we're coming back on another dive.
Oh, that's what we did earlier, right?
Yeah.
That's the stuff.
Yep.
Okay.
Yeah, so this relatively small outcrop is the site of a lot of activity, and one of the unique features of this very small outcrop is the presence of the venting of low temperature fluids that are slightly warmed compared to bottom water, up to about 12 or 15 degrees Celsius, compared to bottom water, which has a temperature of about 2 degrees Celsius.
Bridge.
Control bridge, all copy.
Just the pilot sending a new waypoint to the bridge there.
Sorry about that. There's a bit of a ridge here, so we're having to come up a little bit.
Okay, that's fine. Yeah, my guidance to follow a contour doesn't have to be exact. So as we're coming around here, we might see some additional markers that were left behind on prior expeditions. You can see that there's spots here with higher densities of octopus. We have pretty thoroughly explored this outcrop on previous expeditions, but we haven't yet on this expedition, and it's 9 years later, and it might look a little different. You can see that the surface of the rocks looks a little different to where we were before. It seems that there's more sediment built up in this area. Could give us an indication of the direction of the predominant current and the delivery of particles in the water. We're seeing some more of these kind of more spindly white corals, potentially of the D1 clade. Or not the D1 clade, sorry, that was a different one. Oh, we got a nice swimming octopus. Again, some of these Venus face sponges in the background. A very large coral coming into view up ahead.
Oh, wow.
Extremely large. It's probably a very old coral. For reference, in case anybody's not familiar, you might see that there's two green laser lines right in the center of the camera. Those are set to be 10 centimeters apart. So you can see that this coral is probably, I don't know, 3 meters across.
At least.
At least, maybe more. Incredibly large. Surprisingly, though, it doesn't seem to have many associated animals on it. I was recently on an expedition in the Papahanaumokuakea Marine National Monument, and often in these large fans of corals, you would see lots of brittle stars and other species that climb up onto the coral into the water column. We're seeing less of that here, although there's maybe some. If we were to zoom in, we might see some. Looks like there might be an anemone attached. This appears to be a type of bamboo coral. You can tell by the rings. That's what gives it its characteristic names. Interestingly, it seems that most of the polyps are closed.
Just a smidge more so I can't see those lasers. Perfect. All right, Kim.
Thanks for that. We can keep moving.
Actually,
Oh, and there's a sea cucumber swimming into view. He's like, you thought corals were cool? Check me out. So we're waiting for a ship's move? Is
Yeah,
that what you
the
mentioned?
vessel is moving, but I just getting to an uncomfortable position and wanted to catch up a
Yeah,
little bit.
that's fine. So we can spend more time imaging if we've got time. And this seems like a great thing to follow.
We have laser off.
Yeah.
Okay. Thank you.
So I believe we collected a specimen of this sea cucumber earlier in the dive.
There you go. Wow.
You see that? Is that vibrating?
I'm not sure. I think it's just something attached.
Which one, my lower left?
It's very mesmerizing to watch. Come back down to the seafloor after following our sea cucumber friend up into the water.
This is a much more comfortable position.
Okay.
Continue.
Yes, let's continue, please.
Switched on the modes. If you want to start the vessel kind of northeast.
And we're seeing several more large coral fans coming into view.
Should we put our updated waypoint?
Update the waypoint.
So if you could log biology observation of several different large coral fans, well, actually, and different coral species, and sponges, and a fish. And you can use the different checkboxes for that. Fish, sponges. Oh, that's much better. Thank you. The animal guide is over next to you somewhere. I believe it's a type of cusk eel. There's also a black coral right behind the eel. I don't think that's a rat tail. Rat tail. Yeah. As we're moving along this face, you can see that the rocks also look a bit different than what we've seen before, more sheet flow types, along with these former pillows that are really crusted over. There's some other very large coral fans coming into view here. You can see that they're also kind of oriented in similar directions, so that also gives us an indication of current. How does the current feel down here? Is it pushing us at all?
Yeah, it looks like it's going up the hill. It's not really pushing us, but yeah, you can see it
Yeah.
going from right to left in this view.
More on this side than you saw on the other side?
Say it again?
More on this side than you saw on the other?
Maybe a little, yeah.
Oh, okay.
Saying that, it's still very minimal.
Yeah.
Another one of the yellow sponges coming into view here as we come around the corner. A lot more sediment over on this side.
So bright, I can't no detail out of it.
Yeah. I always love the zoom in shots where you can see that there's so much in that sediment. Tiny little animals. Several different types of sponge species over here, including these Venus-based sponges that we're zooming in on. Surrounding them are different yellow hydroid. And then, of course, the large octocoral fans. If we fly over that large sponge, is there any way to see in it? Yep. Really amazing fabrics that are created by these sponges. Hopefully nobody has vertigo. All right. How are we doing on our ship move?
Let's go. We're clear to
Okay.
move if you're ready.
Yeah, let's go. Here's another black coral coming into view in the bottom right. I believe we collected some black corals earlier in the dive, out in the sedimented abyssal plain.
Yeah, just keep it.
Too far?
It's a nice jelly.
Sure.
So as we're moving onto this east side, where we see more of these corals, we're also seeing less observation of the features that the octopus seem to preferentially attach to. Not seeing as many spots with just the black bare rocks, unsedimented. So there definitely seems to be some habitat differences as we go around this seamount. Another black coral in the bottom of the frame. Some more very large sponges or corals coming into view on the starboard side. And then, again, find one spot with no sediment, and there you find the octopus.
Could be wrong, but it looks a lot bigger than the ones we've seen.
Yeah, if we could spend a moment getting a little closer to interrogate this, just with the video. It looks large. I'm curious if there's any evidence of shimmering water here.
That one is definitely a lot larger than what we've been seeing.
Yeah, it's also sitting in a different position. Many of the ones we've seen before have been with their arms facing outward. This one has its... no, there's nothing that we can open.
Is there a way to mark this spot?
Yep, so we can drop a waypoint in the navigation. Well, it does look like there might be some shimmering back where the arms are attached.
Shimmering between us and it.
Look at this, there's tons of shimmering
Where do you see it?
in this view.
Oh, okay, yeah, in the brow view. Yeah, now I can see it. There's a lot of shimmering coming out of this little hole. Do we have time to sit down here and do a temperature measurement?
Sure, yeah, we need to, let's go and swap the vessel.
Yeah.
And hop into there, what, to keep moving on a little bit. I'm happy with it, yeah. Rich, could you, and I'll stop in that minute just now. Mark, can you turn the lasers on as well? Yeah.
So again, one of the things that we're interested in about this tiny little outcrop in the middle of the Pacific Ocean in Costa Rica's waters is the fact that there's fluid circulation. There's water weeping out of this environment. It's not a hot hydrothermal system. It's a low temperature hydrothermal system. The waters here have been measured up to 15 degrees Celsius. And this seems to be a feature that octopus associate themselves with. And I'm not sure if we've interrogated this spot before. There's no marker here that we've been here. And this octopus looks a bit different than what we've seen on the other side. So with the lasers on, now we can see how big this animal is. So it's at least 10 centimeters across at its head. Thanks for that. And we're going to try to measure the temperature of the water that seems to be leaking out of the seamount here.
Grab the ePro. Yeah. And I'll get this on the right page. There it is. As soon as you grab it, I'll just port you in.
Mm-hmm.
I'm just gonna turn my heading.
And we can turn the lasers off when you have a chance. All right, so now in the picture-in-picture, you can see the reading of the temperature sensor as we're trying to get it back into this area. What's that?
Oh, just the
543, 54.
If they want to touch its arms, there we go, thanks. Thanks, grab my line.
So it's definitely warmer in there, but it's a large area, so it's kind of diffusing.
Yeah, and I'm wondering kind of if we were to come in from that way, but that's gonna be really hard to do with that large.
Yeah, there's a
Yeah.
overhang.
Yeah, exactly. Well, so it's definitely elevated. We've confirmed that. I think the max I saw was 5.6. Let me see. Can you record that as a temperature measurement, please?
5.
Yeah. Okay, you can go ahead and pull the temperature probe out, please. We see that there's, before we pan away, there's a shrimp there that I'd like to zoom in on if I can.
Yep, go for it. You got it. Oh, nice. Got you on the set line.
So this is another indication to us of the warmer waters here with these shrimp that look like vent type shrimp. You can see that there's a lot of shimmering in that water. So it's possible that there is, the waters are warmer than we measured, just based on where we could get that temperature probe. I'd like to try to collect a rock. Do you think that would be possible?
Sure. Let's get this probe stashed. Let's see what we can find.
In the meantime, could we turn the lasers back on?
At some point, not necessarily now, but there were 2 species of shrimp in that
Yeah.
pool. The one that we collected already. Did we collect the little red one?
I don't know.
The one that's like right. You can just
Yeah.
see it at the.
I'm-
It's just something to flag for like later on.
Okay.
It doesn't have to be now, but both of those look very seep and well, the other one is seepy venti, but that one also looks seepy venti like Olivia's. Yeah, the little red one.
It's the toxic response, right?
And that's what. Well,
That's what Emily was saying,
I don't
is that
know.
it's like hypothesized that
Yeah.
they're in a more, it's like a response to toxicity potentially.
I wouldn't think that that's here. These fluids are not going to have high sulfide. That would be toxic
Okay.
in a
Do we
seep
know
type
what-
of environment.
Okay, do we know what the response was? It was like orange eyes and stuff?
No.
The spreadsheet is pretty good, by the way. Looks great. That's a nice view with the lights.
Got the mood lighting going.
Take a frame grab of that. Okay. Also shows the definition of the hydroids here.
She's wanting to find a rock inside there. All that's pretty rigid, but we can
Yeah.
poke around at it to make sure.
Yep. The one I'm interested in is that one. I'm not sure it's going to break off, but it definitely looks like an intact piece. And most of the pieces we've collected so far today on this dive have been rubble.
We're going to ram in. Okay, what do you think about that? Maybe I did too much. Maybe we could just rotate a little bit, like a little bit more.
I think everyone's thinking it's
I take back what I said. That one is open.
And we could use the net and then stick it in there.
Yeah.
It would be great to go for this if we could. Oh, that's a lot of octopus.
That's a lot of octopus for one dive, but we don't have to notify them anymore.
Yeah, I think we should do it.
Before we grab it, can I just get a frame grab, please? You can leave the minute there. That's fine. Thanks. Okay. We ready when you are. So we're interested in the rocks that are in the flow here and the incrustation that's on them. You can see that these rocks are very black, which is an indication that they have coatings of manganese oxides. Those manganese oxides often co-precipitate other metals. It looks like it's pretty well stuck in there. Yeah. Sometimes they're lucky and they break off, but today's not one of those days. Okay, we tried. We can go ahead and stop. So we do have space to try to collect another specimen, and we do have space in the bio box as well. So if we could try to get one of the nets out of the rock box.
One of the nets, I would recommend that one standing out there. And we're gonna try
Oh, that's a terrible shot.
Should signs come in?
just so we're prepared, which bio box is this going into?
It's gonna go in bio box 2. This is the annoying bio box that has that cube floaty in it.
Try and push this against you, just to straighten up. The wire box.
Is this a Octo sample?
Excuse me?
Are you going to the Octo?
Yes.
So yeah, we're gonna try and collect this particular octopus here.
If you want, I can grab the frame, give you a perfect straight on.
Yeah, actually, do that. Rotate it a little bit. Bring it outboard, touch maybe.
For those who are not familiar with how ROV manipulators work, here in the control room, we have little models of each manipulator that are operated by 2 different pilots working together.
So it's gonna really pucker in once it starts to get dark there.
So one of our major objectives of this expedition is to understand the biology of these deep sea octopus, to understand the different species, the different sexes, if they're all the same species or different species. This one has a different, it looks different than some of the others that we've seen. What's that? Oh.
Sweet. Fast. There you go.
Nice. That was great.
Well done.
That was awesome.
Well done, pilots.
And firebox 2 open.
A reminder that there is a marker in there that's going to want to float out. It's fine if it does. We'll figure that out. The biology of deep sea octopus is not well known, and these specimens are incredibly important for understanding their biology and their diversity. It's often difficult to just see an octopus and understand its the sex of the animal, so by having a collection, we can do that. We're also very interested in the microbiomes. Wonderful positioning.
Yeah,
Thank you.
yeah. Well done.
Let's record that. What? Are you ready for the CTD? Yeah. You saw the plan of when it's going to be deployed? Mm-hmm. Oops, what did I just do? Okay, so you're going to wake up for the recovery of this. Okay.
And then I will stay until the deployment because my
Yeah. You don't need to ask permission to go to sleep. It's totally fine. Yeah. Oh, wow. Turn around for 1 minute, and there's a beautiful ctenophore.
Good zoom.
So one of my favorites. Those rainbow colors are just amazing. So we can resume the ship's move if you think we need it to begin
Sure.
following the contour again.
Yeah. Okay. Yeah. Rich, we can resume broadcasting with please.
ROV control bridge. Okay, continuing move to the waypoint.
I think.
No, that's good.
Because we want to be on deck by 11.
I'm just watching the time.
Yeah, that's okay. So we're continuing on our transect around the current contour we're on. We're currently at a depth of about 2,990 meters. You see very steep sides on this end of the outcrop. Just coming around the south side of this feature and starting to head east-northeast. Seems to be another tube anemone off to our left. Or maybe that's just a stain in the rock. Hard to tell with a shadow. Yeah, it's just a stain in the rock. It's not a tube anemone. Just kidding. But again, quite a nice diversity of different glass sponges, different types of octocorals, stoloniferans, crustaceans. What looks to be a rather large sea star at the top of the frame.
Is there anything that strikes you differently since the last time you were here?
Yeah, so when I was last here in 2014, I don't remember the abundance being this high, but I might just not be remembering well. I mean, the most obvious difference between then and now is when we did those prior expeditions in 2013 and 2014, both of which were done in December of those years. We did not observe any viable baby octopus in the egg sacs. And on this expedition, we have not only seen babies in the egg sacs, but also seen juvenile octopus swimming out of their eggs. So they're definitely here. The other notable difference is the last time I was here, I was in the Alvin submersible. So this was literally outside the window as opposed to 3 kilometers underwater, which was pretty incredible.
And I think we come across some Alvin plates.
We have.
Yeah.
Yes. It's an important part of figuring out buoyancy, bringing a little extra weight with you. You can see a lot of, as we zoom in on these rocks, not only do we see sediment, we see debris from dead sponges. So that kind of brownish material that's collected on sponges. You'll see some live ones there, the white ones. Very steep cliff face. So on this side of the outcrop, we're definitely seeing fewer examples of where the fluid leaks out of the seafloor. That's similar to what we saw in 2013 and 2014, that most of that was on the southwest side of the outcrop. You see some of these incredibly large fans of coral, like the one in the top left, that indicates this animal has been around for a long time. Corals grow relatively slowly. This is probably several hundreds of years old, if not older. Mike, now that we're out of the heat of the moment, just commenting again on your amazing netting skills. That's not an easy task. Yeah, so that's a yellow, some type of yellow sponge. Has its Alex, I'm curious what our audience is like today on this dive. Can you?
Yeah, I was just about to look that up.
Thanks to everybody that's tuning in. This is the second dive of the Octopus Odyssey Expedition. We're exploring the Dorado Outcrop. This is a site we've been to before. On tomorrow's dive, we'll be visiting an outcrop that's somewhat similar in size to Dorado Outcrop and also has elevated heat flow measurements that have been done. So our goal is to go visit this totally unexplored seamount to see what we find, and we're very curious to know if we're going to find octopus at that location. This is a beautiful, beautiful jelly. On tomorrow's dive, which will probably begin around 10 a.m. Costa Rica time, noon on the U.S. East Coast. You can join us for that. We'll probably be on the seafloor around noon Costa Rica time. And you can join us as we explore.
Yeah, our audience today pretty similar to the audience yesterday.
Okay. Our
Definitely
weekend warriors.
Yeah, you can certainly recognize some names, right?
Yeah.
So we do have some pretty faithful audience members that show up routinely. But it's always fun to see where things spike up. Like, for example, yesterday we got a big spike, when we first started finding the octopus.
Sure.
Yeah.
Well, thanks for tuning in. If you want to pop in the chat and let us know where you're from, it's always fun to know where our audience is listening from.
Have someone asking for a little blue water treat. I don't know that we're going to get the
look
blue
at that.
water today.
We've got another little guy floating into our camera.
Oh, yeah.
Looks like another Tina 4. So for those interested in tuning into tomorrow's dive, just to give you a preview, we're going to be actually starting out in the abyssal plain for a few hours, doing heat flow measurements and also doing some transects of the animals that live in the abyssal environment. And we'll do that for a few hours, and then we'll move on to the outcrop and start exploring and seeing what we find. So if you want to stay tuned to the Facebook feed and others to find out when we're on the seafloor and follow along with us. Oh, here's another beautiful sea cucumber. Is this a sea cucumber? What is this? Yeah. It's a very full sea cucumber. You can see its digestive tract in there. It is little. Wow, we've got an audience coming from all over, from Australia to the United States, covering so many different time zones. Thanks for tuning in. That's how I need to start moving around. I think so, yeah. I think so.
Captures my feelings for the day.
A rather large crinoid coming into view.
Yeah.
Another black coral off on the left side.
So there is considerable amount of current here.
Oh, we're feeling some current here.
And it is lateral, not so much going up the hill anymore, it's now pushing us sideways.
Okay, so coming from the north and pushing to the south. Yeah.
Yeah, and you can see that fan of coral is facing that instead of the other ones were facing across the elevation.
We have one of these yellow sponges that we sampled earlier on this dive.
Disaster averted.
Yes, of course. And so what I can do is I will start because you're putting this into this one. I can go into CMOD and start putting these in, if that's helpful. So if you put those in, and then I'll move the rest in here. Well, not wrong, because it'll go into both. Because yes, SOI did. So this is
actually increased my power a little bit.
Okay.
It's
all samples taken, and it doesn't have like the ethanol formula numbers.
Wow. You could see that it was going to be steep on the map, but this is just a wall.
I can't tell from this angle, but usually it's fine.
Okay.
crevice, I wonder if we'll see anything here.
Yeah.
Yeah, right? Nice little fracture in there.
Done putting in the hard type of sheet. I will then go and get all the positions and put them into here, and then I can copy everything from here and put it into this one.
That's a weird one.
So that's great. Thank you.
Yep, so the geology observation, fissure is one of the features.
Ooh, it goes under there. Yeah. Big star there?
Yeah, big sea star on the left side.
Wow.
Is it possible to change the lights a little bit so it looks a little blown out in the view?
Yeah, if I'm looking looking up, that's the problem.
Okay,
I can
okay.
look back down,
I see, okay, that's
is
great.
that better?
Yeah,
I mean, you can see stuff blowing out of like the top of this fissure, you know, the current right at the top. Is that just us now?
No,
Oh, okay.
that is the current.
Just west through the gap.
Yeah.
So that was the top of the ridge, that's a crack. Just hoping we see something cool in there.
really steep slopes here. It's likely at the base of this there'd be a lot of rubble. The question about whether we're seeing eggs attached to the rocks here, probably not. That's probably not what we're seeing. It's probably stoloniferan that are in those kind of stringy white masses.
We got some big fans coming. Wow.
Yeah, so some really large fans coming into view here. If we could bring the lasers back on when we get closer to those, so
Yeah,
we
I
can.
see that, thanks, I'll change the course now.
Please. Just so we can get an estimate for the size of this fan. So those lasers you see are just at the base of that coral. Those are 10 centimeters apart. This is probably another 3-meter width coral. Really large.
Currents, once again, going right up this ridge.
Yeah.
Okay.
The direction that thing's facing.
Looks like there's an anemone attached to this coral.
Because that shrimp just whipping. Another picnic on it?
Yeah. A couple different attached species on this coral.
Yeah.
See all the polyps kind of blowing in one direction with this
Yeah, some
current.
stocked barnacles. They picked a good spot.
You can also see that the anemone's tentacles are all blowing backwards. It's really quite an exceptional current.
I'm still confused if we're autofocusing. It seems, try it the other way, it seems, sometimes it goes backwards.
You can see the current with the particles moving into this coral here.
That's definitely manual. That's okay. Yeah, there you go. All right, go ahead.
You can see that part of this coral has probably been predated on because it's just branches with no polyps attached. One of the primary predators are sea stars. Hi. We have more in this.
I think we can put it on the small branch of that one.
Of this? You think, okay, you would like a, it might be difficult because the current here is really tough. So stopping and having a stable
The
position to
sponge
sample
just
might be
right off of the
difficult.
screen.
But I think
Just
we've seen
barely
other
hanging on.
species, So maybe we find it again.
Yeah. Corn dog in the wind.
This is so big.
It's really big. Yeah. All right, we can keep going. I think we've interrogated this one.
It's fully embarrassed.
Nice sheet flow structures on this rock here at the top. tells us a little bit about the lava type as it was coming out.
Yeah.
So we're right near the top of this outcrop at about 2,940 meters.
I'm going to toss up the map.
Remarkably different biology at the top here, and that there's far less of it. And you see it really seems to like where the current is, down on the sides.
I'll toss up the map one more time just so
Okay.
we can see that big flat top to it.
Yeah, exactly. So you can see the little yellow icon is the indication of Sebastian and where we are in relation to that flat top and how steep those sides are, as imaged with the AUV Sentry back in 2013. We collected multi-beam data of this region, which we will work to get into seabed 2030 as soon as we can. And yeah, wow. It's like night and day going up on the top versus what we were seeing on the sides here. So yeah, if we could drop back down as we're moving along, that'd be great. Can we zoom out on the nav map, please? Just so I can see where we are. Yeah, so this is actually if you were to just keep lateraling to the northwest, that's kind of that's the heading I want, northwest.
Yeah, that's kind of what I'm doing, but as you can see, it
Yep.
still waiting.
Exactly. We've got about another hour of bottom time before we'll have to end this dive. There is a black coral there, yes. Bathypathes. What do you think, Wendy?
This is stuff. It's showing 21.
We can turn the lasers off. Yes? You want to try to get them? Yeah, okay. I know you just called in a ship's move, if we could maybe pause it so we can try to do a sampling.
Actually, I'd like him to catch up some.
Okay.
Keep him going for a minute, and then I will definitely pause when he's good.
Okay. Yes? Yep, we will. So we're going to try to sample the Bathypathes first? Yeah? Okay. So this black coral species. The only place we have left to put stuff is the slurp. So what, in the past I've seen grab it with a manipulator and then bring the slurp in with the other one, or bring it to the slurp chamber, the nozzle, and then open the manipulator and let it go. How do you feel about that?
Yeah, it sounds right. We'll get it out, see if you can hand me the nozzle, and I'll just hold it, you grab it. That's basically what she's saying. I'll get it on the port side.
Well, you may not even need to take it off the basket.
Yeah, it's really tough to see. But
Okay.
Let's take a look. See, yeah, see, it's.
Oh yeah, it's kind of down there. Yeah.
This must be for rocks, but they don't have samples in them. That these are not available right now.
No.
I said, know what that stands for.
They have the nets. They have a net and something like that, and it would not stay in there.
Oh,
It
it's
would
not.
stay
I didn't know what it
we
was.
have now. I do it this stuff. Yep.
If you want that one, and he's gonna hand you the nozzle.
No, the current's pushing us.
Yeah, I'm trying. something?
We
I
did.
think it was filled with, yeah, some, basically, because it,
So we need to.
We wanted to put that crab in it, but it didn't, or not the crab, but the spider,
Yeah,
and
you
it didn't
never
go in
did.
there. So it's got some sediment in it, but I think that's okay.
Okay, perfect.
Yeah, I don't want to have to reach around and try to push it again.
Just try to grab that. Maybe we want to tell all the real estate, not draws. This is about. We could just grab it loosely around the nozzle of each. Turn anywhere we're not. Just close it very slowly. Yeah. Up to you there, I think. Let it go. Okay, rotate that to where it's pointing kind of up. That rotates that. Just take off to the side a little bit, right off to the side of the picture. You got to try and hold still.
So for our audience at home, we have 3 pilots working on trying to arrange this sample collection. Takes a lot of teamwork. We have a lot of current pushing the ROV right now, so we have one pilot free flying to try to keep us in place, balance those thrusters to keep us from moving. Another pilot operating the starboard manipulator with the red fingers is going to try to make the collection. And then a third pilot holding the port side manipulator with the suction sampler. Because right now, our only storage on the vehicle for biological samples is the suction container.
Section's going. Might take it more towards the roof now, like, just be aware. You got a gravy in there. Then blowing. Once you get closer, hopefully we can overpower the current.
Maybe you should just push it into the current, let the current blow it in.
Why, take your jaws just a little bit so it's pointing up, so I can see it in that camera as well. Clockwise. Clockwise rotate on your wrist. Yeah, it's good. Yeah.
Can you take some photographs? Just as a nice contrast.
Try flying forward. Yeah, just too much. All the way in there. Come on. Come on.
Yeah.
Getting blown, the rock thing's behind me and I'm trying to fight it. I think it'll be like just hard enough on it. Got the ankles right there. Yeah. So we'll just go there. I need to spin around and get some kind of blue outside. Yeah, it's also hard like this stuff. Is that? Your wrist a little shake. If it's strong enough. Yeah, if it's strong enough. Like volume or something. Yeah, just
Yep.
Alright, why don't you swap the nibs again, get it with something, cause you can, with the handle, I think you can do a better job shaking it if you rotate a little bit again. And then go outboard a touch. Yeah, that's it. If you start putting your arm out the way, yeah, an arm go. Yeah, that's better. This is for helping cameras, it's helpful.
Yeah. It's a 4-person operation here. 8 hands.
Get in there.
Yeah, I think, once it gets into that ribbed area, it's probably stuck. But maybe it'll stay in the slurp sample on the way back.
Straight shot. Yeah, you're starting to get some debris in there. So it's in the tube somewhere.
Yeah, it's in the tube somewhere. That'll be fun. Okay, so for this one, record that the sample is in the slurp sampler tube. It's not, and supposed to be in chamber 8. Either way, we have about 40 minutes, and we're just gonna keep exploring. Yeah. Okay. Okay.
I think that's better than new clothes.
Yeah. Okay.
But Maria and Rachel can
Yeah.
substitute
Yes.
Holly, no?
Mhm.
But if you want to
Just keep
switch,
going,
you want
shoulder up a lot, and shoulder left.
I'm good here.
You guys haven't really done a shift though.
We haven't done a shift.
Yeah, no, they should.
Just talk practice before, so you run out of trouble in the dummy arms.
Yeah, what they didn't do. I picked it up.
And I was my friends, so kind of with bumpers, they kind of.
I haven't gotten to do it much.
It would also be helpful because I need to talk to Rachel about her back plan for tomorrow.
Okay.
While we're finishing up, yeah, because I saw that you put the files in there. But we need to.
Gotta know. I want to talk about it.
Yeah, this was the last one. Yeah. Oh, this was from when we tried to get the
All right, so we have about 40 minutes left to continue exploring. Wow, look at that sea star. That is a huge, beautiful large sea star there.
This is actually the same location, so it has nothing These notes normally aren't there. You'll see also.
Here's another good example of the strength of the current. You can see the crinoids.
We got just an eel down in there?
Yeah, every group is kind of done it. Like, for me, she kind of like stick to the writing because she's her. Yeah, the computer is like, you just, I got it. Yeah. I mean, I don't figure it out. It's the hardest part is reading the animals. Yeah, I know, I was really happy to start.
That's all changed their head as well. Yeah, this is gonna start getting good stuff.
Yeah.
Okay.
So watch for like a second.
Because of the drop off, sorry.
No, the vessel is fighting the current and the winds as well, so it's
Okay.
got its heading best for them. And if I continue to come around this, eventually, like, I'll be on the wrong side of the boat.
Right. What about, would it work, which way is the boat wanting to go? What is it fighting? Because we can also just come across the top.
This your preferred method?
The issue is me being toward the front of the boat.
Yeah.
Our safe spot is off the
heading
side.
to the
Gotcha.
current, if that works for you.
And yeah, it's kind of closing the angle on our launch point as we're coming around this seamount. It would be better if we could have the 90 on, but obviously that's maybe not good for.
We could also drop down and back to the end of the ship, if that's okay.
Yeah, I mean, if you need us to change the heading, that's fine. We can do it. We'll just put a bit more power on and some extra thrusters. And what you want is the head more to the south. Is that right?
That's right. Yeah, that's correct.
They're gonna help us out.
Yeah.
Okay. Yeah, we can do that.
Yeah, just so we're a bit more perpendicular with our launch point.
Copy that.
Awesome. Yeah, this book does do that. Yeah, they're helping us out. That's great.
Looks like some broken coral here, which is not surprising since it was so steep up above with a very strong current. So looks like some of those pieces have broken off and fallen down. Part of the fun of deep sea exploration around seamounts is you really have to think about vehicles in 3 dimensions, where the ship is, what currents and wind the ship is experiencing on the surface of the ocean, and then where the vehicle is on the seafloor, what currents it's experiencing, what currents the tether is experiencing, and then what is the shape of the seafloor you're trying to look at. So you really have to imagine yourself down here. So this looks like a sheet flow, again covered with sediment from the current. That's a very large pink crinoid attached to that coral. Shift is over. Thanks. Thanks, Holly. I'm glad we got some good samples for you.
Yeah, I'm excited. It's gonna be a long night.
Hi. Hello. So you haven't done this yet, right? Okay, so the first thing you want to do is log out and log in as you.
2 adults from yesterday, and we have like 3 different batches of eggs. One of them has like 60 juveniles, and then those are going to count as one.
What is that? That looks like a, what is that? Can we zoom in on that a little closer? I don't think it is, no. I think it's a type of jelly that has been like impaled on this coral. Unknown. So go to biology observation, not sample. No, Excel. Biology observation. Coral and unknown. And then just, if you want to describe it, you can, or you just estimate. Okay, we can.
I thought that was a sea cucumber.
Yeah.
It's a little soft for that one.
Yes.
Alright, so
Also, the black of it gives me an indication that it's maybe not healthy. We have another what I think is a cusk eel and another black coral coming into view. So which is just fish. You can also do geology observations if you want. Sheet flow. So we've gotten a little quiet on the microphone here as we have a shift change going on and some new folks getting their first shifts on a dive. So getting some training on how we record information here in the control room. Joining me on data logging is Dr. Rachel Lauer from the University of Calgary. She's an expert in geophysics and is part of helping us understand fluid flow in this area. Her team leads the heat flow measurements that we do. Also on the shift with me is Dr. Maria Isabel Sandoval, a geologist from the University of Costa Rica, who's interested in paleoceanography and studying the sediments here. A little shrimp there.
ROV Bridge. Yeah, how should I say it? A few minutes.
Yeah, it's better. That works, thank you.
So here in the frame, we have several large fans of octocorals. Can we pivot to the left real quick? I want to interrogate, to just look with the camera at this yellowish sediment. What is that? Not sure why the rocks here have this golden yellow color. Okay, we'll see if we see that again. You can keep going. Thank you. There's a nice little purple sea star right in this, almost in the center of the camera. hard to get a shot without any gear in it. Guess that's
Just
okay.
zoom in a tiny bit and it should clear. There you go.
Can we look at these little white hot dogs on the seafloor? Oh, they're sponges. Never mind. I thought they were some kind of moving animal, but I think they're just blowing in the current. A type of stalked sponge. It looks like a sea cucumber. Wow, even zooming in, you can see the current. That's great. Thanks. Yep, looks like an octopus on a slow walkabout. So for our audience at home, we have about 20 more minutes to continue exploring the seafloor on this dive before we begin our recovery. We anticipate launching our next ROV dive. Oh, there's that yellow sediment again. Launching our next dive tomorrow around 10 a.m. Costa Rica time, getting on bottom around noon Costa Rica time, UTC -6 for those not in Costa Rica. And we'll be exploring a new environment tomorrow. We'll be going to an unnamed outcrop just a little southwest of this region where we think that we have another type of Dorado outcrop, a place where water is coming out of the seafloor. We're going to go try to understand if there's also octopus in that environment. It's nice to have a visitor here. Another type of eelfish. Okay. There's seems to be a lot more fish on this side.
Yes, it is. The frame, the head frame, no. Yeah, it's no.
Here's more of that discolored rock you were looking at.
Yeah.
See a little better, maybe.
Yeah. Yeah, can we hover down there? Almost looks like carbonate or a carbonate-rich mud with this manganese crust on top. I seem to remember seeing this before when we were here in 2013 and 2014. Remember that, Rachel? Yep. Yeah, okay.
I think that yellow spot was the same.
Yeah, I think so too. It is a sponge.
Sometimes the web page doesn't update, it'll take what you put in, but it won't show down here, and you just have to refresh the page. And sometimes it forces you to log out, so sometimes you have to log out. So it does. So when they were spinning, it was fun. There's times when it forces you to log out, you won't have this in left. So you'll know that you won't have it, so you're not losing anything.
Do it on the northwest. Not too far. Yeah, roughly there.
There's our ROV, we got an updated waypoint.
Yeah, copy that.
Whee!
No wonder, I'm like, you're going left, I'm going right.
Yeah,
Oh boy.
that's fine. Wow, just look at that. So many sponges and corals. How does the current feel here?
It's still relatively high. Maybe not as bad as before, but
Okay. Well, definitely some good food delivery here based on how many animals are here. Yes?
So the next slide is just basically
It's a similar feature like this. It's just a tiny little, but we're actually not sure because we don't have good multibeam here.
so
Yeah, so Rachel gave me some waypoints, I just need to get them to you. Yeah.
For a couple of lines.
Yep, exactly. I was gonna do that as soon as this dive was over, or as soon as we're off bottom.
Okay, right, thanks. I didn't do it.
The edge of the cliff.
We just have to back down this ridge for a while.
I think there's a comment for you. Can you see it here?
I don't want to pass this ridge.
Do you want to say anything? Yeah, come on over here.
Yeah, you don't want to do that. Until I get the vessel a little bit further north.
So joining me in the room is my colleague, Dr. Maria Isabel Sandoval. She's going to say a few words.
Hola, saludos a todos. Hola, mami. Aquí muy contenta que esté conectada viendo las observaciones del fondo oceánico a 3.000 metros de profundidad. Sí. Y lo que vemos ahí son octocorales encima de algunas pillow lavas con esponjas cristalinas de tipo Venus. Bueno, un saludo y un abrazo a todos.
We have just a few more minutes to go here on the seafloor as we explore a part of the outcrop that we haven't yet seen on this expedition. Much larger and more dense octocoral fans than we were seeing on the southwest side of the outcrop. Josh, are you able to put up the picture-in-picture of the Fledermaus view? Thank you. So on the screen here, the picture-in-picture view is showing an exaggeration of the seafloor bathymetry. You can see the location of Sebastian. On the side of the seamount, you can see where we were before. It was extremely steep. We're still in a pretty steep area. We're heading towards the northwest. We won't quite make it all the way to the end on this dive, but definitely seeing a part of the seamount we haven't yet seen. Yeah, thanks so much, Josh. So as we're dropping down here on the seamount, we're now at about 3,020 meters, getting into a good position for starting our recovery. On the midship side, Again, for anybody that's tuning in just now, we only have a few more minutes on the seafloor for this dive before we recover. We've got lots of samples on board that we'll be processing through the night. We will be starting our next ROV dive tomorrow morning, 10 a.m. Costa Rica time, and probably on the seafloor around noon Costa Rica time tomorrow, exploring a environment that's never been visualized before. We'll be bringing that to you live here. This seems to be a different type of coral. I don't think I've seen this yet. No, the white part behind it. And yes, the yellow is a crinoid. Behind it is a type of coral. It's a really lovely contrast of colors there. Looks like there's a little anemone in the background off to the left. It's a type of cup anemone that's a little bit retracted, I think. I'm not an expert, but I think that's what it is. It's okay. There's an eel off to our right. Definitely a considerable number of fish, eel fish on this side of the seamount. You can see some blocks that have rolled off the seamount here. You can tell that this piece has probably been knocked off the seafloor, off the outcrop for quite a while because it has some very large coral attached to it. Indicates quite a lot of longevity, so zoom in here real quick. If I'm not an expert, but I think that is a type of tunicate down here off to the right of the crab. Oh, there's another one. Interesting little translucent organisms. Lots of Venus face sponges here as well. Different types of sponges. Looks like some demo sponges also in the background. It's really incredible diversity considering we're at 3,000 meters. See several fish in the background. If you were tuned in yesterday, you got a really unique experience seeing some fish predation occurring in the deep sea. Often you have to put out baited traps to see something like that. Oh, we have two water column animals here. Looks like a modified shrimp and a jelly in the background. Both of these are relatively small. Nice starfish in the background too.
Put that right in the center and I'll just try to hold it.
The camera? Okay, will do. I won't touch it.
No, no, no. You're doing me a lot. It's not like that at all.
Yeah. No, I know what you're trying to do. Wow, look at the detail on that. Incredible. What a great show for the end of our dive. Thank you to everybody that's been tuning in. We're going to be ending our time on the bottom here quite soon. But this is just a spectacular way to end our visit to the bottom of Dorado Outcrop. Another extremely successful dive of the Octopus Odyssey Expedition. Great. All right. Thank you to the pilots. Expert manipulation as expected and as always.
All right, you ready to recover?
We are.
Okay.
Yeah.
So let's go.
Yeah, would you like to?
Did you cut it already?
No, we're still live. If they would like to say goodbye in Spanish. Okay, yes, please.
Bueno, muchas gracias a todos los que nos acompañaron hoy. Esperamos que nos acompañen mañana en otra interesante excursión al fondo del mar. Hoy estuvimos entre 2.000 y 3.000 metros de profundidad y mañana vamos a estar en otro lugar que nunca hemos estado antes, entonces va a ser una gran experiencia. Muchas gracias por habernos acompañado de nuevo y probablemente vamos a empezar a transmitir mañana a partir de las 12 del mediodía. Hasta luego todos.